Hey guys, tonight we're doing something a little different, a little darker, a little more unsettling. We're diving into the biology that keeps scientists awake at night, the concepts so strange, so counterintuitive, so fundamentally weird that they challenge everything you thought you knew about life itself. Forget the textbook diagrams of cells and DNA helixes you memorized in high school. Tonight, we're exploring the shadows, the biological paradoxes that make researchers pause and question their own understanding, the evolutionary nightmares that reveal nature's indifference to our comfort, the microscopic horrors and macroscopic mysteries that prove the living world is far stranger and far more disturbing than any horror film could imagine. So, before we begin this 3-hour journey into the uncomfortable corners of biology, take a moment to like the video and subscribe, but only if you genuinely enjoy what we do here. And let me know in the comments where you're tuning in from and what time it is for you. I always love seeing our global community of curious minds gathering in the quiet hours, seeking knowledge in the darkness. Now, settle in, dim the lights, maybe turn on that fan for some white noise. Get comfortable in your bed, because we're about to spend the next few hours together exploring concepts that will reshape how you see yourself, your body, and the living world around you. Because tonight, we're not just learning biology, we're confronting it. We're looking directly at the parts of life that textbooks gloss over, the realities that make us uncomfortable, the truths that sit in the back of every biologist's mind like unwelcome guests at a dinner party. We'll meet cells that refuse to die decades after their host has been buried. We'll discover that the bacteria living inside you outnumber your own cells and might be influencing your thoughts right now. We'll explore proteins that act like contagions despite not being alive. We'll confront the reality that your body is constantly choosing which parts of itself to sacrifice and consume. These aren't abstract concepts happening in distant laboratories. This is happening inside you, right now, as you listen. Your cells are dividing, dying, and betraying their programming. Your mitochondria are carrying out ancient instructions written in DNA that isn't even yours. Your immune system is fighting battles you'll never consciously know about. And somewhere deep in your tissues, senescent cells are accumulating, refusing to die but no longer truly alive. Some of tonight's concepts will challenge your sense of identity. Others will make you question what it means to be an individual organism at all. A few might genuinely disturb you, and that's exactly the point. Because the most profound understanding comes not from comfortable facts, but from truths that force us to see ourselves differently. And I promise you nothing will look quite the same when we're done. So let's begin our descent into the beautiful, terrible complexity of dark biology. We begin with a woman named Henrietta Lacks. She died on October 11th, 1951, at the age of 31. Cervical cancer had ravaged her body, spreading through her organs until nothing could be done. She was buried in an unmarked grave in Virginia, mourned by her family, forgotten by the world, except for one small problem. Henrietta Lacks never really died. 8 months before her death, a doctor at Johns Hopkins Hospital took a sample of her cancerous cells without her knowledge or consent. This was common practice in 1951. Patient consent wasn't really a thing yet, especially not for a poor black woman in segregated Baltimore. The cells were handed to a researcher named George Gay, who had been trying for years to keep human cells alive in culture. Every sample he'd tried had died within days. Human cells, it turned out, didn't want to live in laboratories. But Henrietta's cells were different, disturbingly different. They didn't just survive, they thrived. They doubled every 20 to 24 hours, growing with an aggression that shocked everyone in the lab. Gay named them HeLa cells, using the first two letters of her first and last name, and then he did something that would change medicine forever. He started giving them away. Within months, HeLa cells were in laboratories across the country. Within years, they were worldwide. They were used to test the polio vaccine, saving countless lives. They were sent into space to see how cells behaved in zero gravity. They've been used in cancer research, AIDS research, gene mapping, and toxicity testing. They help develop in vitro fertilization and cloning techniques. More than 75,000 scientific papers have been published using HeLa cells. If you stacked all the HeLa cells ever grown, they would wrap around the Earth at least 3 times. Their total mass would weigh more than 50 million metric tons. Think about that for a moment. Henrietta Lacks died in 1951, but her cells, the ones cut from her cervix without permission, are still alive, still growing, still dividing in laboratories on every continent. Parts of her have traveled to space. Parts of her exist in medical facilities you've probably walked past. She's been dead for over 70 years, but she's also more alive now than she ever was. So what made HeLa cells immortal? The answer lies in something deeply unsettling about cancer itself. Normal human cells have a built-in limit to how many times they can divide. It's called the Hayflick limit, and we'll explore it more deeply later tonight. But cancer cells don't follow this rule. Henrietta's cancer cells had activated an enzyme called telomerase, which rebuilds the protective caps on chromosomes that normally wear down with each division. Essentially, her cells had figured out how to reset their biological clock. They became, in the truest sense, immortal. But here's where it gets philosophically disturbing. These cells aren't Henrietta. They're cancer cells that killed her. They're her body turned against itself. Cells that stopped cooperating with the organism and started pursuing their own survival at any cost. And they succeeded. While Henrietta died, her cancer achieved a kind of immortality she never asked for and never knew about. For decades, the Lacks family had no idea that Henrietta's cells were still alive. No one asked their permission. No one told them their mother, their wife, their sister was being bought and sold by medical supply companies. HeLa cells became a multi-million-dollar industry, and the Lacks family didn't see a cent. Some of her children couldn't even afford health insurance, while their mother's cells were generating enormous profits worldwide. The ethical nightmare doesn't stop there. In the 1970s, researchers contacted Henrietta's family, not to inform them or compensate them, but to take their blood samples. They wanted to study the family's genetics to better understand HeLa cells. The family, with limited education and no understanding of what was happening, gave consent without fully knowing what they were agreeing to. Their genetic information was published without their knowledge. Their privacy was violated in the name of science. Today, guidelines have changed. Informed consent is now required. Patients have more rights over their biological materials. But none of that helps Henrietta Lacks, whose cells continue to divide in laboratories where her name is known, but her humanity is often forgotten. She's become more laboratory tool than person, more acronym than woman. There's something deeply haunting about biological immortality achieved through disease. Henrietta's cells live on because they're the thing that killed her. They're not a legacy she chose or a gift she willingly gave. They're a violation that happened to result in scientific progress, and that progress is undeniable. Millions of lives have been saved because of HeLa cells. Medical advances that seemed impossible became routine. But at the center of that progress is a woman who never consented, never knew, and never benefited. Her story forces us to confront uncomfortable questions. What does it mean to be alive when parts of you continue existing after your death? If your cells are growing in laboratories around the world, are you somehow still here? And if biological immortality is possible, but only through the lens of cancer, what does that say about the relationship between life, death, and disease? Henrietta Lacks wanted to live. She had 5 children, a husband, a life she wasn't ready to leave, but her body had other plans. And now, more than 7 decades after her death, her cells keep dividing, keep growing, keep existing in a strange twilight state between person and property, between life and laboratory specimen. The next time you hear about a medical breakthrough, there's a decent chance healer cells were involved, which means there's a decent chance a piece of Henrietta Lacks contributed to it. A woman who died in segregated Virginia is now part of the foundation of modern medicine, not by choice, not with consent, but undeniably, permanently, and somewhat disturbingly, forever. So Henrietta Lacks is dead, and yet, in the most literal biological sense, she's more alive than almost anyone who's ever lived. And that paradox, that uncomfortable truth, is where dark biology begins. You've probably heard that you're unique, one of a kind, an individual. But here's the uncomfortable truth that microbiologists have been quietly discussing for decades. You're not an individual at all. You're a colony, a walking ecosystem, a biological democracy, where your own cells are barely in the majority. For years, scientists claimed that bacterial cells in your body outnumbered human cells by 10 to 1. That number has been revised recently to closer to 1 to 1, but that's not exactly comforting. Imagine standing in a room where half the people are you and half are complete strangers. Now imagine those strangers have their own agendas, their own survival strategies, and their own ways of influencing what happens in that room. That's your body. Right now. These aren't invaders. They're residents. You were born relatively sterile, but within hours of entering the world, bacteria began colonizing your skin, your mouth, your gut. By the time you were 3 years old, you'd established a microbiome, a complex community of trillions of microorganisms that would live with you for the rest of your life. They're in your intestines, on your skin, in your mouth, nose, and even your eyes. Everywhere your body interfaces with the outside world, bacteria have set up shop. And here's where it gets disturbing. They're not passive passengers. They're active participants in your biology. Your gut microbiome, that dense jungle of bacteria in your intestines, weighs between 1 and 2 kilograms. That's roughly the same weight as your brain. And increasingly, scientists are discovering that these 2 organs, if we can call the microbiome an organ, are in constant communication. It's called the gut-brain axis. Your gut bacteria produce neurotransmitters, the same chemicals your brain uses to send signals. They make serotonin, dopamine, and gamma-aminobutyric acid, also known as GABA. In fact, about 90% of your body's serotonin, the chemical most associated with mood and happiness, is produced in your gut. Not by your cells, by bacteria. So when you're feeling anxious, depressed, or inexplicably happy, how much of that is you, and how much of that is them? When you crave certain foods, is that your body asking for nutrients? Or is it your gut bacteria manipulating your brain to feed them what they want? Because yes, they can do that. Studies have shown that gut bacteria can influence food preferences, sending signals to your brain that manifest as cravings. Let's sit with that for a moment. You think you're choosing to eat chocolate or craving a burger. But there's a non-zero chance that the trillions of bacteria in your intestines are chemically nudging your brain toward decisions that benefit them. They want sugar, they want fiber, they want specific nutrients, and they've evolved ways to make you want them too. This isn't science fiction. This is documented, peer-reviewed science. Researchers have transplanted gut bacteria from obese mice into lean mice, and the lean mice gained weight. They've transferred bacteria from anxious humans into germ-free mice, and the mice developed anxiety-like behaviors. Your microbiome doesn't just digest food. It influences your metabolism, your immune system, your mood, and possibly even your behavior. There's also evidence, still preliminary but deeply unsettling, that your microbiome might influence your personality. Studies have found correlations between certain bacterial populations and traits like sociability, stress response, and even risk-taking behavior. If your gut bacteria can produce neurotransmitters and send signals to your brain, then the line between you and them becomes uncomfortably blurred. And it gets weirder. Your microbiome is unique to you, like a fingerprint. The specific combination of bacterial species you carry is shaped by your genetics, your diet, your environment, your early childhood, even how you were born. Babies delivered vaginally acquired different bacteria than those born via cesarean section, and those differences can persist for years. You've been collecting bacteria your entire life, and they've been shaping you just as much as you've been shaping them. But here's the part that should really unsettle you. When you take antibiotics, you're not just killing the infection, you're carpet bombing your entire microbiome. Beneficial bacteria die alongside harmful ones. And when your microbiome is disrupted, the consequences can be severe. Antibiotic use has been linked to increased rates of obesity, diabetes, inflammatory bowel disease, and even mental health disorders. Because when you kill off the bacteria that were helping regulate your metabolism and mood, you're not just treating an infection, you're disrupting an ecosystem. And that ecosystem can be hijacked. Clostridium difficile, often called C. diff, is a bacterium that causes severe, sometimes fatal diarrhea. It's normally kept in check by other gut bacteria, but when antibiotics wipe out the competition, C. diff can take over. The treatment? A fecal transplant. Yes, doctors take stool from a healthy donor and transplant it into the patient's colon to reintroduce beneficial bacteria. It sounds absurd, but it works. Because your gut bacteria aren't just passengers. They're your defense system. Scientists are now exploring the idea that many modern diseases, things like autoimmune disorders, allergies, and metabolic syndrome, might be diseases of the microbiome. Our sanitized, antibiotic-heavy, ultra-processed modern lifestyle has altered the bacterial communities we evolved alongside for millions of years. We're cleaner than ever, but our immune systems are confused. Our microbiomes are depleted, and the consequences are showing up in rising rates of chronic illness. There's even research suggesting that your microbiome affects how you respond to medications. Cancer patients with certain gut bacteria respond better to immunotherapy. Antidepressants work differently depending on your microbiome composition. The same drug can have wildly different effects in different people, and increasingly, scientists believe the microbiome is part of the explanation. So who are you really? Are you the human cells, the ones with your DNA, Or are you the whole system, the superorganism made up of human and bacterial cells working in uneasy collaboration? When you make a decision, is it your neurons firing, or is it the result of bacterial signals influencing those neurons? Where does you end and they begin? Philosophy aside, the practical implications are staggering. You're not just a body, you're an environment. And like any environment, you can be healthy or degraded, balanced or invaded, thriving or collapsing. Every meal you eat, every antibiotic you take, every stressful day you endure, you're not just affecting yourself. You're affecting trillions of other organisms that are, in turn, affecting you. The idea of the individual, the autonomous self making free choices, gets a lot more complicated when you realize you're outnumbered in your own body. You're not a person, you're a population. And the scariest part? You're not even the majority shareholder. So, the next time you think a thought or feel an emotion, just remember, there's a decent chance it's a group decision. You think you know who you are. You've got your DNA, your genes, your biological blueprint that makes you uniquely you. Except there's a problem. You might be carrying someone else's DNA. Not metaphorically, literally. Cells with completely different genetic code living inside your body, dividing, functioning, existing as a foreign presence you never invited and probably never knew about. This phenomenon is called chimerism, named after the chimera of Greek mythology, a creature made from parts of different animals, and it's far more common than anyone wants to admit. In fact, if you're a woman who's ever been pregnant, you're almost certainly a chimera. If you're a twin, there's a chance you absorbed your sibling before birth. If you had an older sibling, their cells might be living in your body right now. And if you received a blood transfusion or an organ transplant, well, you're definitely carrying someone else's genetic material. Let's start with the most common form, maternal fetal chimerism. During pregnancy, cells don't just stay on their side of the placental barrier, they cross. Fetal cells enter the mother's bloodstream, and maternal cells enter the fetus. This isn't a malfunction, it's normal. But here's what's unsettling. Those cells don't just visit, they stay for decades. Studies have found fetal cells in mothers' bodies up to 27 years after giving birth. These aren't dormant fragments. They're living, functional cells that have integrated into the mother's tissues. Fetal cells have been found in mothers' brains, hearts, lungs, kidneys, and skin. They can even differentiate into different cell types, becoming neurons, heart cells, or immune cells, depending on where they end up. So if you're a mother, you're carrying pieces of every child you've ever been pregnant with. They're in your bloodstream, they're in your brain, and they're not passive. Some research suggests these fetal cells might help repair damaged tissue. There are documented cases of fetal cells migrating to a mother's injured heart and contributing to healing. Other studies suggest they might play a role in autoimmune diseases, where the mother's immune system starts recognizing these foreign cells as threats. But it goes the other way too. Maternal cells cross into the fetus and can persist for life. You might be carrying cells from your mother that have been with you since before you were born. And if your mother was carrying cells from her previous pregnancies, well, you might also be carrying cells from older siblings you've never met. It's a biological Russian nesting doll of genetic identities. Then there's twin chimerism, which is even stranger. During early development, when an embryo is just a cluster of cells, sometimes two embryos start to form, fraternal twins, each with their own genetic identity. But occasionally, before they fully separate, one twin absorbs the other. not in a violent way, just in a quiet, cellular way. The absorbed twin's cells become incorporated into the surviving twin's body. The surviving twin grows up completely unaware they were once two people. They look normal, they feel normal, but hidden in their tissues are cells with completely different DNA. These individuals are called tetragametic chimeras because they have 4 sets of genetic contributions instead of 2 from each absorbed twin. And sometimes this only gets discovered by accident. There have been legal cases where maternity tests failed because the DNA in a woman's blood didn't match the DNA of her children. She knew she'd given birth to them. Hospital records confirmed it, but the DNA said otherwise. How? Because the DNA in her blood came from the twin she absorbed in utero, while her ovaries carried her own genetic line. She was, genetically speaking, her own twin. The children were biologically hers, but the tests were sampling the wrong her. One of the most famous cases involved a woman named Lydia Fairchild. She applied for public assistance and was required to provide DNA evidence that she was the mother of her children. The tests came back negative. Authorities accused her of fraud, of trying to claim children that weren't hers. She was threatened with losing custody. Only after her lawyer requested that DNA be taken from multiple parts of her body did the truth emerge. The DNA in her blood didn't match her children, but the DNA in her cervical tissue did. She was a chimera, carrying 2 distinct genetic lines in her own body. In another case, a man failed a paternity test for his own son. He and his partner had conceived through in vitro fertilization, so there was no question about biological parentage. Yet the DNA didn't match. Further testing revealed that the man's sperm carried different DNA than his blood. He had absorbed a twin in the womb, and the cells that became his reproductive organs came from that twin. Genetically, he wasn't his son's father. His absorbed twin was. These are extreme cases, but they reveal something deeply uncomfortable about biological identity. We think of ourselves as singular, unified individuals, but at the cellular level, many of us are patchworks, mosaics, living collages of different genetic identities coexisting in one body. And it's not always invisible. Some chimeras display visible differences in their bodies, patches of skin with different pigmentation, eyes of 2 different colors, a condition called heterochromia, hair that grows in distinct color patterns. These aren't just quirks. They're clues that different cell populations with different DNA are expressing themselves in different parts of the body. Chimerism raises philosophical questions that biology alone can't answer. If you're carrying someone else's DNA, are you still just you? If part of your brain is made of cells that came from your child, are your thoughts entirely your own? If you absorbed a twin who never got to live, are you one person or 2? And if someone commits a crime, but their DNA at the crime scene doesn't match the DNA in their blood, who's legally responsible? Is less than #0. 7# is greater. There's also the question of how common this really is. Most people never get comprehensive genetic testing of multiple tissues, so mild chimerism often goes completely undetected. You could be carrying foreign cells your entire life and never know it. Some scientists estimate that up to 8% of fraternal twins might have some degree of chimerism from early embryonic exchange of cells. And nearly all mothers carry fetal cells for decades after pregnancy. So the next time you look in the mirror and think you're seeing you, remember, you might be looking at a collaboration, a biological merger, a body that houses more than one genetic identity, stitched together so seamlessly that you've never noticed the seams. You're not just one person contained in one body. You might be several, and they've been with you all along. The boundaries of self aren't as clear as we'd like to believe. Inside your tissues, in your blood, maybe even in your brain, there could be cells that aren't genetically yours, and yet they're part of you, functioning, surviving, living a quiet existence in a body they helped build or maintain. You're not an individual, you're a collective, and some members of that collective you never even met. Your cells are counting down to death. Right now, as you listen to this, every cell in your body carries a biological timer that's been ticking since the moment you were conceived. And when that timer runs out, the cell stops dividing. It can't be reset, it can't be rewound, it just stops. This isn't a disease, it's not a malfunction. It's a deliberate feature of your biology, programmed into your DNA by millions of years of evolution. You are designed to age. You are designed to die. The timer is called a telomere. Picture the plastic tip on the end of a shoelace, the part that keeps it from fraying. Telomeres are similar. They are protective caps on the ends of your chromosomes, made of repetitive DNA sequences that don't code for anything. They're just buffers, sacrificial segments that exist to be destroyed, and with every cell division, they get a little bit shorter. Here's how it works. When a cell divides, it has to copy its DNA, so each daughter cell gets a complete set of genetic instructions. But the machinery that copies DNA, an enzyme called DNA polymerase, has a problem. It can't quite reach the very end of a chromosome. Every time your DNA is copied, a tiny bit at the end doesn't get replicated. It's like making a photocopy of a photocopy. Each generation loses a little detail at the edges. If your chromosomes didn't have telomeres, this would be catastrophic. Important genes would get chopped off with every division. But because telomeres act as buffers, it's the meaningless, repetitive sequences that get lost instead. Your actual genes stay safe. For a while, the telomeres absorb the damage, shortening a little more with each division. They're dying so your genes don't have to, at least not yet. Scientists have measured this. Human telomeres shorten by about 30 to 200 base pairs with each cell division. They start out around 8,000 to 10,000 base pairs long at birth. Do the math, and you realize your cells can only divide a finite number of times before the telomeres are too short to function. That limit is called the Hayflick limit, named after Leonard Hayflick, who discovered it in 1961. Hayflick was studying human cells in culture, and he noticed something strange. No matter how perfect the conditions were, no matter how much nutrient-rich medium he provided, the cells would divide around 50 times and then stop. Not die, necessarily, just stop dividing. They'd sit there, metabolically active but unable to reproduce. He called it cellular senescence, and it shattered the prevailing belief that cells could be immortal given the right environment. The reason they stopped? Telomeres. Once telomeres get too short, they trigger a cellular alarm system. The cell interprets short telomeres as DNA damage because, in a way, that's exactly what they are. The cell's quality control mechanisms kick in, and the cell either stops dividing or, if the damage looks severe enough, initiates programmed cell death, apoptosis, cellular suicide. The cell dismantles itself from the inside, packaging its components for recycling, and quietly disappears. This is why you age. Your cells have a built-in expiration date. The cells in your skin, your blood, your gut lining, they're constantly dividing to replace old, damaged cells. But every division shortens their telomeres. Eventually, they hit the Hayflick limit and stop. Your tissues lose their ability to regenerate. Your skin becomes thinner. Your wounds heal slower. Your immune system weakens because it can't produce new cells fast enough. You're not falling apart because something went wrong. You're falling apart because everything is going exactly as evolution intended. But here's the disturbing part. There's an enzyme that can rebuild telomeres. It's called telomerase. It adds those repetitive DNA sequences back onto chromosome ends, essentially resetting the clock. And some of your cells have it. Stem cells use telomerase to maintain their ability to divide throughout your life. Germ cells, the ones that become sperm and eggs, use telomerase so they can pass on full-length telomeres to the next generation. But most of your cells, they don't have active telomerase. It's turned off. Evolution could have given you telomerase in every cell. You could theoretically have cells that divide indefinitely, that never hit the Hayflick limit, that never senesce. So why don't you? Because there's a cell type that does exactly that. Cancer cells. Almost all cancer cells reactivate telomerase. That's how they become immortal. They bypass the Hayflick limit, ignore the telomere alarm, and divide without stopping. Telomerase is one of the key differences between a normal cell and a cancerous one. So, your body faces a terrible trade-off. If your cells have telomerase, they can divide forever, but they risk becoming cancerous. If they don't have telomerase, they're protected from unlimited division, but they age and eventually stop working. Evolution chose aging over cancer. It decided that a limited lifespan was better than the risk of uncontrolled growth. Your death is the price you pay for not being made entirely of tumors. And this isn't theoretical. There's a genetic condition called dyskeratosis congenita, where telomerase doesn't work properly. People with this condition have extremely short telomeres. Their cells hit the Hayflick limit much earlier than normal. They age rapidly. Their bone marrow fails because it can't produce new blood cells. Their skin deteriorates, their organs give out, they rarely live past 30. It's a glimpse of what happens when the telomere clock runs too fast, and it's horrifying. On the other end of the spectrum, scientists have genetically engineered mice with extra telomerase. These mice lived longer than normal mice. Their tissues stayed healthier, their cells kept dividing, but they also developed more cancers. The trade-off held. You can have longevity or you can have safety, but not both, at least not with our current biology. Telomere length is now used as a biomarker for aging. People with shorter telomeres tend to die younger. They have higher rates of cardiovascular disease, diabetes, and dementia. Chronic stress shortens telomeres faster. So does smoking, obesity, and lack of exercise. On the flip side, meditation, exercise, and a healthy diet seem to slow telomere shortening. You can't stop the clock, but you might be able to slow it down. There are companies now selling telomerase activators, supplements that supposedly boost telomerase activity to slow aging. The science is shaky at best, and the cancer risk is unknown, but the appeal is obvious. If aging is caused by telomere shortening, and telomerase can rebuild telomeres, then maybe, just maybe, we can hack the system. Maybe we can turn the clock back, or at least pause it. But even if we could, should we? Aging isn't a bug, it's a feature. It's evolution's way of making sure damaged, mutation-laden cells don't stick around forever. It's why children don't inherit their parents' accumulated genetic errors. It's why species can adapt over generations instead of being locked into the bodies of immortal ancestors. Death makes room for the new. It's brutal. It's unfair, but it's also necessary. So every time your cells divide, your telomeres get shorter. Every breath you take, every meal you digest, every thought you think requires cells to divide, to replace themselves, to burn through their limited quota of divisions. You're not just living, you're spending, and the currency is time measured in base pairs, counted down by telomeres that shorten with every tick of the cellular clock. You are a temporary arrangement of matter, held together by chemistry and governed by timers you can't see. Your body knows it's mortal. Your cells know they're expendable. And deep in the heart of every chromosome, the countdown continues. Aging isn't something that happens to you. It's something written into you from the very beginning. Imagine waking up one morning with an overwhelming urge to do something completely out of character, something dangerous, something that goes against every survival instinct you have, and you can't explain why. You just feel compelled. Now imagine that urge isn't coming from you. It's coming from a microscopic parasite living in your brain, rewiring your neural pathways to serve its own reproductive goals. This isn't science fiction. This is happening right now to millions of people, and most of them have no idea. The parasite is called Toxoplasma gondii, and it might already be inside you. Studies estimate that anywhere from 30 to 50% of the global human population is infected. In some countries, the infection rate is closer to 90%. You probably don't know you have it. Most people never show symptoms. But that doesn't mean it's doing nothing. Toxoplasma is a master manipulator, and its primary talent is changing behavior. Let's start with how it works in its natural hosts. Toxoplasma's life cycle is bizarrely specific. It can only sexually reproduce inside the intestines of cats. Any cat, domestic, wild, doesn't matter. But to get there, it needs to infect other animals first. Rodents are its preferred intermediate host. So here's the problem. Rats and mice are terrified of cats. They've evolved for millions of years to avoid the smell of cat urine because that smell means death. It's a hardwired survival instinct. But when a rat gets infected with Toxoplasma, something changes. The parasite migrates to the rat's brain and forms microscopic cysts in specific regions, particularly the amygdala, which processes fear and emotion. And then it does something extraordinary. It removes the rat's fear of cats, not just reduces it, eliminates it. Infected rats are actually attracted to the smell of cat urine. They seek it out. They linger in areas where cats have marked territory. Behaviorally, they're committing suicide, and they don't care. This isn't coincidence. This is precision biological warfare. The parasite needs the rat to be eaten by a cat so it can complete its life cycle. So it hacks the rat's brain, rewires its fear response, and turns a survival instinct into a death wish. The rat gets eaten. The parasite reproduces in the cat's intestines. Its offspring are shed in cat feces, contaminating the environment, and the cycle begins again. It's elegant, it's horrifying, and it works. Now here's the part that should concern you. Humans aren't part of Toxoplasma's natural life cycle. We're accidental hosts. Dead ends, biologically speaking. The parasite can't reproduce if a cat eats a human, but it can still infect us. And when it does, it still migrates to the brain. It still forms cysts in the amygdala and other regions, and increasingly, scientists believe it still changes behavior. The effects are subtle. This isn't mind control in the Hollywood sense. Infected humans don't suddenly start seeking out lions, but the research is piling up, and it's deeply unsettling. Studies have found correlations between toxoplasma infection and increased risk-taking behavior. Infected individuals are more likely to get into car accidents. They have slower reaction times. They show reduced fear responses. Some research even suggests personality changes. Infected men tend to become more aggressive, more suspicious, and less respectful of rules. Infected women tend to become more outgoing, more trusting. Is the parasite causing these changes, or are people with these traits simply more likely to get infected? The causality is still debated, but controlled studies in animals are harder to dismiss. When you infect mice with toxoplasma, their behavior changes predictably. When you treat the infection, their behavior returns to normal. Something is happening in the brain, and it's not just sitting there quietly. There's also a darker connection. Multiple studies have found higher rates of toxoplasma infection in people with schizophrenia. The association is consistent across different countries and populations. Again, causality isn't proven. But toxoplasma cysts are found in the exact brain regions that malfunction in schizophrenia. The parasite triggers chronic inflammation in the brain. It alters dopamine levels, and dopamine dysregulation is a hallmark of schizophrenia. The connections are too strong to ignore. Then there are the suicide studies. Several research groups have found elevated toxoplasma antibodies in people who have attempted suicide, particularly violent suicide attempts. One study found that women with toxoplasma infection were 1.5 times more likely to attempt suicide. Another found that the risk increased with the level of antibodies, suggesting a dose-dependent effect. The more parasites, the higher the risk. Now, correlation is not causation. Toxoplasma doesn't make people suicidal in any simple, direct way. But if the parasite reduces fear responses, increases impulsivity, and alters neurotransmitter levels in the brain, it's not hard to imagine how it could tip someone already struggling toward a tragic decision. The parasite doesn't have intent. It's not trying to harm humans. We're just collateral damage in a life cycle that evolved around cats and rodents. So, how do you get infected? The most common route is through undercooked meat, especially pork and lamb. Toxoplasma forms cysts in muscle tissue. If you eat infected meat that hasn't been cooked thoroughly, you ingest the parasite. The other major route is through cat feces. If you clean a litter box, garden in contaminated soil, or touch anything contaminated with cat feces and then touch your mouth, you can get infected. Once inside you, the parasite is there for life. Your immune system can suppress it, force it into a dormant cyst stage, but it never leaves. Pregnant women are warned about toxoplasma for good reason. If a woman gets infected during pregnancy, the parasite can cross the placenta and infect the developing fetus. This can cause severe brain damage, blindness, and even death. That's why doctors tell pregnant women to avoid changing litter boxes and to cook meat thoroughly. But if you were already infected before pregnancy, the dormant cysts usually don't reactivate. Your immune system keeps them in check. Unless your immune system fails, in people with HIV, cancer, or anyone on immunosuppressive drugs, toxoplasma can reactivate. The cysts rupture. The parasites start multiplying again, and this time they can cause severe, sometimes fatal, brain inflammation. It's one of the most common opportunistic infections in AIDS patients. The parasite that's been sitting dormant in your brain for decades suddenly becomes lethal when the immune surveillance drops. But toxoplasma isn't the only parasite that manipulates behavior. There's a fungus called Ophiocordyceps that infects ants. It grows inside the ant's body, and when it's ready to reproduce, it releases chemicals that compel the ant to climb to a high point clamp its jaws onto a leaf, and stay there. Then the fungus erupts from the ant's head, releasing spores to infect more ants below. The ant becomes a zombie, a vehicle for fungal reproduction, and it has no choice. There are parasitic worms that infect crickets and make them jump into water where the worms can reproduce. There are barnacles that infect crabs, chemically castrate them, and make them care for barnacle larvae as if they were their own eggs. Nature is full of parasites that hijack nervous systems, alter behavior, and turn hosts into puppets. And humans, with our complex brains and our confidence in free will, are not exempt. So the next time you make a decision, especially a risky one, ask yourself, is this really me? Or is this something living in my brain, pulling strings I can't see, nudging my behavior toward outcomes that benefit it, not me? The line between self and invader is thinner than you think. And for millions of people carrying toxoplasma, that line might already be blurred. You like to believe your thoughts are your own, that your choices come from some essential you, untouched by outside influence. But if a microscopic parasite can change a rat's deepest survival instinct, what makes you think your brain is any less hackable? Toxoplasma doesn't need your consent. It just needs access. And once it's in, it's there forever, quietly shaping who you are in ways you'll never consciously notice. Cancer isn't an alien invasion. It's not a virus from outside or a bacterium you caught. It's you, your own cells. Cells that carry your DNA, your genetic fingerprint, your biological identity. Except somewhere along the way, they stopped following the rules. They stopped cooperating with the rest of your body. They decided to survive at any cost, even if that cost is killing the organism they're part of. Cancer is betrayal from within. It's evolution happening inside you, and you're losing. To understand cancer, you need to understand what holds your body together in the first place. You're made of roughly 37 trillion cells. That's 37 followed by 12 zeros, and every single one of those cells is, technically, an independent living organism. It has its own metabolism, its own machinery, its own capacity to survive and reproduce, but it doesn't. It cooperates. It follows instructions. It divides when told to divide, stops when told to stop, and dies when told to die. This cooperation is what makes you possible. Your body is essentially a cellular society. Skin cells, liver cells, neurons, blood cells, they all have specialized jobs. They work together. They communicate through chemical signals. They sacrifice their own interests for the good of the whole. It's not altruism. It's programming. Every cell carries the same DNA, the same instruction manual, and part of that manual says cooperate or die. Most cells follow the manual, but occasionally one doesn't. That cell has accumulated mutations, changes to its DNA caused by radiation, chemicals, viruses, or just random copying errors during cell division. Most mutations are harmless. Some are fatal to the cell, but a few, a very unlucky few, give the cell an advantage. Maybe it stops responding to growth-suppressing signals. Maybe it ignores the signal to stop dividing. Maybe it disables its own self-destruct mechanism. One mutation usually isn't enough. But if the cell keeps dividing and accumulating more mutations, eventually it hits a critical threshold. It becomes cancerous. And here's the disturbing part. Cancer is evolution in fast forward. The mutated cell starts dividing uncontrollably. Its descendants inherit the mutations. Some descendants acquire new mutations. The ones that grow fastest, evade the immune system best, and resist damage most effectively survive and multiply. The ones that don't die off. It's natural selection happening inside your body at the cellular level, and the cancer cells are winning because they're optimized for one thing: survival. Not your survival, theirs. Cancer cells are, in a way, reverting to an ancient state. Multicellular life is a relatively recent invention, evolutionarily speaking. For billions of years, life was single-celled. Every organism was on its own, competing for resources, dividing as fast as possible. Then, about 600 million years ago, cells started cooperating, forming colonies, and eventually complex organisms like you. But that ancient single-celled programming is still in your DNA. It's just suppressed. Cancer is what happens when those suppression mechanisms fail. The cell remembers how to be selfish. Your immune system is supposed to catch this. It's constantly surveilling your body, looking for cells that are damaged, infected, or behaving abnormally. When it finds them, it destroys them. But cancer cells are tricky. They're not foreign invaders. They're you. They carry your DNA. So your immune system doesn't always recognize them as threats. And cancer cells evolve ways to hide. They display surface proteins that say, "Don't kill me." They secrete chemicals that suppress immune activity. They build protective environments where immune cells can't reach them. It's an evolutionary arms race, and the cancer is adapting faster than your immune system can respond. Even more disturbing, cancer cells can hijack your body's resources. They send out chemical signals that trigger blood vessel growth, a process called angiogenesis. New blood vessels grow directly into the tumor, feeding it oxygen and nutrients. The cancer essentially builds its own supply lines. It diverts resources away from healthy tissue and uses them to fuel its own growth. Your body is actively helping the thing that's killing it. Cancer cells also lose their specialization. A liver cell is supposed to do liver things. A lung cell does lung things. But cancer cells stop caring about their original job. They dedicate all their energy to dividing. They become generic, simplified, stripped down to the bare essentials of growth and survival. In this way, they're more primitive than normal cells, less complex, more efficient, and far more dangerous. One of the hallmarks of cancer is its ability to metastasize. Most cells are anchored in place. They stick to their neighbors and to the extracellular matrix around them. But cancer cells lose this stickiness. They break away from the primary tumor, enter the bloodstream or lymphatic system, and travel to distant parts of the body. When they land somewhere new, they start growing again. A breast cancer cell ends up in the lung and starts a new tumor. A colon cancer cell ends up in the liver. Metastasis is why cancer is so lethal. A localized tumor can often be removed, but once cancer spreads, it's nearly impossible to eliminate. Here's the terrifying math. Every day, your cells divide trillions of times. Every division is a chance for a copying error. Most errors are corrected by proofreading enzymes. Most mutations that slip through are harmless. But with trillions of divisions happening constantly, the odds catch up. It's not a matter of if a cell will accumulate dangerous mutations. It's a matter of when, and how well your immune system catches it before it becomes a problem. Age is the biggest risk factor for cancer, and now you know why. The longer you live, the more cell divisions you've accumulated. The more divisions, the more chances for mutations. The more mutations, the higher the probability that some cell, somewhere, will hit that critical combination that turns it cancerous. Aging isn't just about telomeres shortening or tissues wearing out. It's about the statistical inevitability of your own cells betraying you. Then there are the external factors: smoking, radiation, certain chemicals. They don't cause cancer directly. They increase mutation rates. They damage DNA faster than your repair mechanisms can fix it. Every cigarette is rolling the dice. Every sunburn, every exposure to carcinogens. You're increasing the odds that a cell will acquire the wrong mutation at the wrong time. And once it does, evolution takes over. Some cancers are driven by viruses. Human papillomavirus, or HPV, causes most cervical cancers. Hepatitis B and C cause liver cancer. Epstein-Barr virus is linked to certain lymphomas. These viruses insert their own DNA into your cells, and sometimes that insertion disrupts important genes. Tumor suppressor genes get turned off. Growth-promoting genes get turned on. The virus doesn't mean to cause cancer, it's just trying to reproduce. Cancer is a side effect, collateral damage. There are also inherited mutations. BRCA1 and BRCA2 are genes involved in DNA repair. If you inherit a faulty version, your cells are worse at fixing damage, mutations accumulate faster, your lifetime risk of breast or ovarian cancer skyrockets. You're born with a head start toward cancer, and there's nothing you can do about the genetic hand you were dealt. You can reduce other risk factors, but the baseline vulnerability is already written into your DNA. Treatment is an evolutionary problem too. Chemotherapy and radiation kill rapidly dividing cells. That's why they target cancer. But they also kill healthy cells that divide quickly. Hair follicles, gut lining, bone marrow. The side effects are brutal. And worse, cancer cells can evolve resistance. If even a few cancer cells survive treatment, they're the ones with mutations that made them resistant. They multiply, and now you're dealing with a cancer that no longer responds to the drugs that almost killed it. It's natural selection under therapeutic pressure, and it's why cancer often comes back. So cancer isn't a foreign attacker, it's a civil war. It's your own cells abandoning the social contract, reverting to selfish, ancient behavior, and multiplying without regard for the consequences. It's evolution optimizing for cellular survival at the expense of your survival. And the cruelest part? The cancer cells don't win either. When you die, they die too. They're trapped in the same body, they're just too short-sighted to care. They're optimized for growth, not long-term survival, and in the end, their success is their extinction. Every cell in your body is one mutation away from treason, and the only thing stopping it is a complex system of checks, balances, and surveillance that's constantly working behind the scenes. Most of the time, it works, but sometimes it doesn't, and when it fails, you're fighting a war against yourself, against cells that used to be on your side, but have forgotten they were ever part of a team. There's something out there that isn't alive, but it can kill you. It has no DNA, no RNA, no metabolism, no cells. It doesn't breathe, eat, or reproduce in any conventional sense. You can't kill it with antibiotics, antivirals, or disinfectants. You can incinerate it at temperatures that would melt steel, and it survives. You can blast it with radiation that would sterilize a surgical room, and it remains infectious. It's called a prion, and it's one of the most terrifying things in all of biology, because it shouldn't be possible, and yet it is. A prion is a protein, just a protein. Specifically, It's a misfolded version of a normal protein that exists in your brain right now. The normal version is called pre-RP, short for prion protein, and every mammal has it. It sits on the surface of neurons, probably helping with cell signaling and copper metabolism, though its exact function is still debated. But normally, pre-RP is folded into a specific three-dimensional shape. That shape determines its function. Proteins are like origami. The way they fold dictates what they do. But sometimes pre-RP misfolds. Instead of its normal structure, it twists into a different shape. This misfolded version is stable, resistant to breakdown, and has one horrifying property. When it touches a normal pre-RP protein, it can force that protein to misfold too. Shape, corrupting shape. It's not reproduction, not in the traditional sense. There's no genetic material being copied. It's more like crystallization. One misfolded protein templates the misfolding of another, which templates another, which templates another. And once the process starts, it doesn't stop. The misfolded proteins clump together, forming aggregates that accumulate in brain tissue. These clumps are toxic to neurons. Cells die. Holes appear in the brain tissue, giving it a sponge-like appearance under a microscope. That's why prion diseases are called spongiform encephalopathies. Your brain literally turns into a sponge, and there's no treatment, no cure, no way to stop it. Once the misfolding cascade begins, it's a death sentence. The most famous prion disease is mad cow disease, technically called bovine spongiform encephalopathy. It emerged in the United Kingdom in the 1980s, when cattle were fed rendered remains of other cattle. Some of those cattle had misfolded prion. When healthy cattle ate the contaminated feed, they became infected. The prions survived the rendering process because heat doesn't destroy them. The cattle developed neurological symptoms: aggression, loss of coordination, tremors, then death. Humans who ate meat from infected cattle developed a variant form of Creutzfeldt-Jakob disease, or CJD. It starts with psychiatric symptoms: depression, anxiety, hallucinations. Then come the physical symptoms: loss of coordination, difficulty walking, involuntary movements, memory loss, dementia, and then, inevitably, death. Usually within a year of symptom onset. Autopsies reveal brains riddled with holes and plaques of misfolded prion protein. The damage is irreversible. But mad cow disease isn't the only prion disease. There's classic Creutzfeldt-Jakob disease, which occurs sporadically in about 1 in a million people worldwide. No known cause. It just happens. A protein misfolds spontaneously, and the cascade begins. There's also familial CJD, caused by inherited mutations in the pre-RP gene. If you're born with a mutation that makes your prion protein slightly unstable, you're at risk of it misfolding sometime in your life, usually in your 50s or 60s. Then there's fatal familial insomnia, one of the most disturbing prion diseases. It's caused by a specific mutation in the prion protein gene. Symptoms usually start in middle age. First, you have trouble sleeping. Insomnia gets progressively worse. Then come hallucinations, panic attacks, rapid weight loss. Your autonomic nervous system starts failing. You lose the ability to regulate body temperature, blood pressure, heart rate, and you stop sleeping entirely, completely. Your brain loses the ability to enter sleep states. Within months, you're dead. There's no treatment, no way to force sleep. The prions have destroyed the parts of your brain responsible for sleep regulation. Another prion disease is Kuru, found among the Fore people of Papua New Guinea. In their traditional funerary practices, they consumed the bodies of deceased relatives, including the brain. If the deceased had Kuru, the prions spread to those who ate the tissue. Kuru means trembling in the Fore language. Victims developed uncontrollable shaking, difficulty walking, and emotional instability. They laughed inappropriately, hence Kuru's nickname, the Laughing Death. Once the Fore stopped practicing mortuary cannibalism in the 1960s, Kuru cases declined. But even today, decades later, occasional cases still appear, because prions can incubate for 50 years or more before causing symptoms. That's the other nightmare about prions. The incubation period can be extraordinarily long. You can be infected today and not develop symptoms for decades. And during that time, you have no idea you're carrying a lethal, misfolded protein in your brain. You feel fine, but the cascade has started. It's just moving slowly, very slowly, until suddenly it's not. Prions challenge our understanding of infection. We're used to thinking of infectious agents as living things. Bacteria, viruses, fungi, parasites. They have genetic material, they reproduce, but prions don't. They're just proteins with the wrong shape. They're not alive. You can't kill them because they were never alive to begin with. Normal sterilization procedures don't work. Autoclaving, the process of using high heat and pressure to sterilize surgical instruments, doesn't fully inactivate prions. Hospitals have had to develop special protocols for instruments used in brain surgery on patients with suspected prion disease. Some instruments are simply incinerated because decontamination isn't reliable. Even more disturbing, prions can cross species barriers. Mad cow disease jumped from cattle to humans. Chronic wasting disease, a prion disease affecting deer and elk in North America, is spreading rapidly. So far, there's no confirmed case of it jumping to humans, but researchers are worried. If it does, we could be facing a new epidemic, and we'd have no way to stop it. Prions in the environment are incredibly persistent. They can remain infectious in soil for years, possibly decades. The mechanism of prion propagation is so simple, it's almost elegant. No need for DNA or RNA to encode instructions. Just a shape that forces other proteins into the same shape. It's infectious geometry. And because the misfolded protein is so stable, it resists all the cellular machinery designed to break down damaged proteins. Your cells have quality control systems that identify misfolded proteins and destroy them. But prion proteins evade those systems. They aggregate faster than the cell can clear them. There's also evidence that prion-like mechanisms might be involved in other neurodegenerative diseases. Alzheimer's disease is characterized by plaques of misfolded beta-amyloid protein and tangles of misfolded tau protein. Both spread through the brain in patterns that suggest a prion-like propagation. One misfolded protein templates the misfolding of others. Parkinson's disease involves misfolded alpha-synuclein spreading through brain tissue. These aren't technically prion diseases because they're not infectious between individuals. But the underlying mechanism, protein misfolding spreading from cell to cell, is disturbingly similar. This raises a terrifying question. If protein misfolding can propagate itself, if shape can corrupt shape without any genetic material involved, how many other proteins might be capable of this? Are prions rare anomalies, or are they just the tip of an iceberg we're only beginning to understand? Could there be misfolded proteins spreading through your brain right now, too slowly to cause symptoms yet, but building toward a threshold? Scientists have tried everything to stop prions: drugs that stabilize the normal protein shape, compounds that prevent aggregation, antibodies that bind to misfolded proteins. Nothing has worked in humans. A few experimental treatments have shown promise in cell cultures or animal models, but translating that to human patients has been futile. By the time symptoms appear, too much brain damage has already occurred. And even if you could stop new prions from forming, you can't repair the holes already eaten into the brain. So prions remain an unsolved nightmare, a reminder that life and death, infection and disease, aren't always defined by the presence of living organisms. Sometimes all it takes is a shape, a fold, a protein twisted the wrong way, touching another protein and another, until your brain is riddled with holes and your thoughts dissolve into static. No virus, no bacteria, just geometry corrupting itself one molecule at a time. And the worst part? You probably have the normal version of prion protein in your brain right now. It's there, folded correctly, doing whatever it's supposed to do. But all it would take is one spontaneous misfold, one random twist in the wrong direction, and the cascade could begin. It's not likely, but it's possible. And that possibility, that silent potential for self-corruption, is sitting in your neurons, waiting. Your cells know how to die, not by accident, not from injury or infection. They know how to deliberately dismantle themselves from the inside, packaging their components neatly for recycling and vanishing without leaving a mess. It's called apoptosis, and it's happening inside you right now. Thousands of cells are committing suicide every second. It's not tragic. It's necessary, because without programmed cell death, you'd be a shapeless blob of uncontrolled tissue. Or worse, you'd be dead from cancer years ago. Apoptosis is one of the most fundamental processes in multicellular life. It's how your body sculpts itself during development, maintains order during adulthood, and removes damaged or dangerous cells before they become threats. And it's eerily precise. When a cell receives the signal to die, it doesn't just explode. It doesn't spill its contents and trigger inflammation. It dies cleanly, methodically, almost politely. Here's how it works. The death signal can come from outside the cell or inside. External signals might be a lack of growth factors, meaning the cell is no longer needed. Or it might be a message from an immune cell saying you're infected, time to die. Internal signals come from the cell's own monitoring systems. DNA is too damaged to repair. Mitochondria are failing. Something's wrong, and the cell knows it. Either way, once the decision is made, the execution is swift. The cell activates a cascade of enzymes called caspases. These are molecular scissors that cut specific proteins inside the cell. Once activated, caspases start dismantling everything. The cytoskeleton, the internal scaffolding that gives the cell its shape, gets chopped up. The cell shrinks and rounds up. The nucleus, where DNA is stored, begins to break apart. The DNA itself gets fragmented into neat pieces. Meanwhile, the cell membrane starts blebbing, forming bubble-like protrusions as the cell loses structural integrity. But here's the key. The cell membrane stays intact. All of this destruction is happening inside a sealed container. The cell packages its remains into small vesicles called apoptotic bodies. These are like tiny garbage bags, wrapped in membrane, containing fragments of the cell's former contents. Nearby immune cells, particularly macrophages, recognize signals on the surface of these apoptotic bodies. Eat me signals. The macrophages engulf the dying cell's remains and recycle the components. No mess, no inflammation, no collateral damage. The cell disappears as if it never existed. This process is essential during development. When you were an embryo, your hands started as flat paddles. You had webbing between your fingers like a duck, but the cells in that webbing received a signal to undergo apoptosis. They died in precise patterns, carving out the spaces between your fingers. Without apoptosis, you'd still have webbed hands. The same thing happens with your feet, your facial features, even the hollow tubes of your circulatory and digestive system. Your body was sculpted by billions of precisely timed cell deaths. Even your brain was shaped by apoptosis. During fetal development, your nervous system produces far more neurons than you'll ever need. Then, in a massive pruning event, about half of them die. The ones that make the right connections, that receive survival signals from their target cells, are allowed to live. The ones that don't, die. It's brutal, but it's how your brain wires itself. Neural connections are refined through selective death. In adulthood, apoptosis maintains balance. Your body produces about 200 billion new cells every day just to replace the ones that die. Red blood cells, gut lining cells, skin cells, they're constantly turning over. Old cells are removed by apoptosis to make room for new ones. Your gut lining replaces itself every few days. Your skin surface cells die and flake off constantly. Apoptosis keeps this turnover orderly. Without it, dead cells would accumulate, clog tissues, and trigger chronic inflammation. Your immune system also relies on apoptosis. When immune cells, like T cells, are no longer needed after fighting an infection, most of them undergo apoptosis. This prevents the immune response from spiraling out of control. But sometimes this system fails. If immune cells don't die when they're supposed to, they can start attacking your own tissues. That's autoimmunity. Diseases like lupus, rheumatoid arthritis, and type 1 diabetes are partly caused by immune cells that refuse to undergo apoptosis when their job is done. Apoptosis is also your primary defense against cancer. Every day, cells accumulate mutations. Most are harmless. But some mutations, if left unchecked, could turn a cell cancerous. Fortunately, cells have internal monitoring systems. If DNA damage is detected, the cell can try to repair it. If the damage is too severe, the cell activates apoptosis. Better to die than risk becoming a tumor. This is why the p53 gene is so important. It's often called the guardian of the genome. When p53 detects DNA damage, it can halt the cell cycle and trigger apoptosis. If p53 is mutated and non-functional, damaged cells don't die. They keep dividing. That's how many cancers start. Cancer cells are, in many ways, cells that have learned to ignore apoptosis signals. They disable the pathways that would normally trigger programmed death. They overproduce proteins that block caspase activation. They ignore signals from the immune system. Essentially, they've become immortal at the cellular level because they've turned off their own self-destruct mechanisms. That's why cancer is so hard to treat. You're fighting cells that refuse to die even when they should. Chemotherapy and radiation work, in part, by trying to force cancer cells into apoptosis. They damage DNA or disrupt cell division so severely that the cell's internal alarms go off. If the cancer cell's apoptosis machinery is still somewhat functional, it dies. But if those pathways are completely broken, the cell survives. That's one reason why some cancers are resistant to treatment. The cells have evolved to be resistant to death itself. On the flip side, too much apoptosis is also a problem. Neurodegenerative diseases like Alzheimer's, Parkinson's, and ALS involve excessive neuronal death. In these conditions, neurons undergo apoptosis prematurely, possibly due to accumulated damage, inflammation, or misfolded proteins. Once those neurons are gone, they're not replaced. Your brain doesn't regenerate most neurons. So when apoptosis is triggered inappropriately in the brain, the result is progressive, irreversible decline. There's also a related process called necroptosis, a programmed form of necrosis that looks messy, like accidental cell death, but is actually controlled. And autophagy, which we'll explore more later, where cells digest parts of themselves to survive. But apoptosis is the cleanest, most controlled form of death. It's death as an act of cooperation. The cell sacrifices itself for the good of the organism. What's philosophically unsettling is that every cell in your body carries the instructions for its own death. Embedded in your DNA are genes that, when activated, will systematically dismantle the cell from within. It's like every cell is born with a suicide note already written, waiting for the right signal to be read. Most cells never receive that signal, but the capacity is always there, dormant, ready. And sometimes, cells die for no clear reason. Spontaneous apoptosis. A perfectly healthy cell suddenly activates its death program. Maybe a signaling pathway misfired. Maybe random molecular noise triggered the wrong cascade. The cell dies, is cleared away, and replaced. You never notice. But it's a reminder that life at the cellular level is precarious. One misplaced signal, one errant enzyme activation, and the self-destruct sequence begins. Your body is in constant negotiation with death. Not the death of the organism, but the death of individual cells. Every moment, your cells are deciding whether to live or die based on signals, damage assessments, and molecular votes. Most choose life. But some choose death, or have death chosen for them. And that's normal. That's healthy. Because uncontrolled life, cells that never die, is cancer. And uncontrolled death, cells that die too readily, is degeneration. The balance between apoptosis and survival is what keeps you alive. Too little cell death, and damaged, mutated, or infected cells accumulate. Too much, and essential tissues break down. Your body walks this tightrope every second of every day. Thousands of cells dying, thousands more being born. The turnover is invisible, silent, and absolutely essential. So the next time you look at your hand, remember that it was carved from a paddle of tissue by precisely timed waves of cell death. The spaces between your fingers exist because cells died to make them. Your brain works because half its neurons were eliminated before you were born. You are, quite literally, shaped by death. And every day, that process continues. Cells die so you can live. It's not morbid. It's just biology. But it does make you wonder. In a body where death is so carefully orchestrated, where does life really end and death begin? Inside every one of your cells, there's something that doesn't belong to you. It has its own DNA, separate from the DNA in your nucleus. It reproduces on its own schedule, it has its own evolutionary history, and billions of years ago, it was a completely independent organism that invaded one of your ancestors' cells and never left. It's called a mitochondrion, and you inherited it solely from your mother. Your father contributed half your nuclear DNA, but your mitochondria, those came exclusively from the egg. which means you're carrying the genetic legacy of an ancient infection that became the most successful symbiosis in the history of life. To understand mitochondria, you need to go back roughly 1.5 to 2 billion years. Life on Earth was still mostly single-celled. Some cells had nuclei, the early ancestors of all complex life, plants, animals, fungi. These cells were called eukaryotes, but they had a problem. They weren't very efficient at producing energy. Meanwhile, there were bacteria floating around that were extremely good at extracting energy from oxygen. These bacteria had evolved a process called aerobic respiration, which generates far more energy than older methods like fermentation. One day, a eukaryotic cell either engulfed one of these bacteria or the bacterium invaded the cell. Normally, the cell would digest it, or the bacterium would kill the cell. But neither happened. Instead, the bacterium survived inside the cell, and the cell kept the bacterium alive because it was useful. The bacterium was producing energy, lots of it, and sharing it with the host. In exchange, the host provided the bacterium with protection and nutrients. It was a mutually beneficial arrangement. And over time, the bacterium became so integrated into the cell that it could no longer survive on its own. It became an organelle, a mitochondrion. This event, called endosymbiosis, is one of the most important moments in the history of life. Without it, complex multicellular organisms like you wouldn't exist. You need enormous amounts of energy to power a brain, to move muscles, to maintain body temperature. Mitochondria make that possible. They're often called the powerhouse of the cell, a phrase you probably remember from high school biology. But it's true. Mitochondria generate adenosine triphosphate, or ATP, the molecule your cells use as energy currency. Almost all of your ATP comes from mitochondria. But here's the eerie part. Mitochondria still have their own DNA. Not much, just 37 genes in humans, compared to the roughly 20,000 genes in your nuclear DNA. Most of the mitochondrian's original genome has been transferred to the nucleus over evolutionary time. But mitochondrial DNA, or mtDNA, still exists, floating in a loop inside the mitochondrion, just like bacterial DNA. And it's passed down exclusively through the maternal line. When a sperm fertilizes an egg, the sperm contributes its nuclear DNA, but its mitochondria are left outside or destroyed. Only the egg's mitochondria make it into the embryo. This means you can trace your mitochondrial lineage back through your mother, your mother's mother, your mother's mother's mother, in an unbroken chain stretching back thousands of generations, all the way to a woman scientists call mitochondrial Eve. She wasn't the only woman alive at the time, probably lived around 150,000 to 200,000 years ago in Africa. But she's the most recent common ancestor of all living humans. Through the maternal line, every mitochondrion in every human alive today descended from hers. But mitochondria are more than just evolutionary relics. They're essential to nearly every aspect of your physiology. They don't just produce energy, they regulate calcium levels, which affects muscle contraction and nerve signaling. They play a role in cell signaling and apoptosis, that programmed cell death we discussed earlier. In fact, mitochondria are often the ones that trigger apoptosis. When mitochondria become damaged, they release proteins that activate the cell's self-destruct sequence. If the powerhouse fails, the whole cell goes down. And mitochondria do fail. They accumulate damage over time. Mitochondrial DNA mutates faster than nuclear DNA because it's not as well protected. There are no histones, those protective proteins that wrap around nuclear DNA. and mitochondria are constantly exposed to reactive oxygen species, or ROS, which are byproducts of energy production. It's like working in a factory where the exhaust fumes slowly poison you. Mitochondria generate energy, but in the process, they damage themselves. This damage accumulates with age. Older mitochondria are less efficient. They produce less ATP and more ROS. Cells with damaged mitochondria struggle to meet their energy demands. This is one of the leading theories of aging. Mitochondrial dysfunction contributes to muscle weakness, cognitive decline, and increased susceptibility to disease. Your cells are powered by ancient bacteria, and those bacteria are wearing out. There are also diseases caused directly by mitochondrial mutations. Because mtDNA is inherited maternally, these diseases pass from mother to child. Mitochondrial myopathies cause muscle weakness and exercise intolerance because muscles need huge amounts of ATP. Leber's hereditary optic neuropathy causes sudden vision loss because the optic nerve's energy demands can't be met. Lee syndrome causes progressive neurological decline in infants and children. These diseases are devastating and currently incurable because you can't easily replace or repair mitochondrial DNA. Interestingly, the number of mitochondria in a cell varies wildly depending on the cell's energy needs. A skin cell might have a few hundred. A liver cell, which is metabolically very active, might have 1,000 or more. A muscle cell, especially heart muscle, is packed with mitochondria because muscle contraction requires constant ATP. Your heart never stops beating, which means your cardiac cells are burning through energy relentlessly. And when mitochondria can't keep up, you get heart failure. Mitochondria also have a fascinating relationship with the immune system. Because they're descended from bacteria, they still carry molecular signatures that resemble bacterial components. Under normal circumstances, this isn't a problem. But if mitochondria are damaged and their contents leak into the cell or bloodstream, your immune system can recognize those bacterial-like molecules and trigger inflammation. There's growing evidence that mitochondrial dysfunction contributes to chronic inflammatory diseases and even autoimmune conditions. Your immune system might be attacking remnants of an ancient infection that's been part of you for billions of years. Then there's the strange phenomenon of mitochondrial heteroplasmy. Not all your mitochondria are genetically identical. Mutations can arise in individual mitochondria, and because each cell contains hundreds of mitochondria, you can have a mix of normal and mutated versions. If the proportion of mutated mitochondria crosses a certain threshold, the cell can't produce enough energy, and symptoms appear. But because the distribution of mutated mitochondria is random, the severity and symptoms of mitochondrial diseases can vary wildly, even within the same family. Scientists are now exploring mitochondrial replacement therapy. In cases where a woman carries severe mitochondrial mutations, doctors can take the nucleus from her egg and transfer it into a donor egg with healthy mitochondria. The resulting embryo has nuclear DNA from the parents, but mitochondrial DNA from the donor. It's been called three-parent babies, which is technically accurate but misleading. The mitochondrial DNA donor contributes only 37 genes, a tiny fraction compared to the 20,000 from the nuclear DNA. But it raises ethical and philosophical questions. Is the donor a parent? What does it mean to inherit genetic material from three people? There's also research into boosting mitochondrial function to slow aging. Exercise, caloric restriction, and certain supplements like NAD precursors are thought to improve mitochondrial health. Some scientists are even experimenting with mitochondrial transplants, injecting healthy mitochondria into damaged tissues. The results are preliminary, but the potential is staggering. If you could repair or replace failing mitochondria, you might delay aging, prevent degenerative diseases, and extend healthy lifespan. But here's the unsettling takeaway. You're not entirely human. At the most fundamental level, your cells are collaborations between two ancient lineages. Your nuclear DNA is eukaryotic, the descendant of cells that evolved nuclei billions of years ago. But your mitochondria are bacterial, the descendants of a microbe that was absorbed and never escaped. You're a hybrid, a fusion organism. And every cell in your body is a reminder that individuality is an illusion. You're a cooperative venture, and the partnership started long before you were born. You inherited your mitochondria from your mother, who inherited them from hers, in a chain that stretches back to the origin of eukaryotic life. Your father's mitochondria, the ones his mother gave him, ended with him. They weren't passed on. This matrilineal inheritance means mitochondrial DNA can be used to trace ancestry in ways nuclear DNA can't. It's a genetic thread connecting you to every maternal ancestor you've ever had. So the next time you feel tired, remember, it's not just you that's exhausted. It's the billions of ancient bacteria living inside your cells, churning out ATP, accumulating damage, and slowly failing. You're only as strong as your mitochondria, and they're only as strong as the evolutionary bargain struck nearly 2 billion years ago. You didn't choose this partnership, but without it, you wouldn't exist at all. Your DNA is not your destiny. That old idea that your genes determine who you are and what diseases you'll get is crumbling under the weight of new science. Because sitting on top of your DNA, like annotations in the margins of a book, are chemical markers that decide which genes get read and which get ignored. These markers can be changed by your environment, your experiences, your diet, even your thought. And most disturbing of all, they can be passed down to your children and grandchildren. Trauma your grandmother experienced, starvation your grandfather survived, the stress your parents endured, all of it might be coded into your biology right now. You're not just carrying your own history, you're carrying theirs. This is epigenetics, literally meaning above genetics. Your DNA is like a massive library containing every instruction for building and running your body. But not every book in that library is open at the same time. Some are locked away, some are bookmarked. Some are actively being read, while others collect dust. Epigenetic markers are the librarians deciding what gets accessed. They don't change the text of the DNA itself, they change whether that text gets expressed. The most common epigenetic modification is DNA methylation. Picture a methyl group, a small cluster of one carbon atom and 3 hydrogen atoms, attaching to specific spots on your DNA. When methyl groups accumulate on a gene, that gene is effectively silenced. The cellular machinery can't access it to make proteins. The gene is still there, intact, but it's not being used. Methylation is like putting a book on a high shelf. It's not gone. It's just unavailable. Another major mechanism is histone modification. Your DNA isn't just floating loose inside the nucleus. It's wrapped around proteins called histones, like thread around spools. How tightly the DNA is wrapped determines whether genes can be accessed. Chemical modifications to histones, acetylation, methylation, phosphorylation, change how tightly the DNA coils. Loose coils mean genes are accessible. Tight coils mean they're locked away. It's dynamic, constantly shifting in response to signals from inside and outside the cell. Here's where it gets unsettling. These epigenetic markers respond to your environment. When you're stressed, your body releases cortisol. Chronic cortisol exposure can alter methylation patterns in genes related to stress response, inflammation, and even immune function. If you experience severe stress, especially early in life, those changes can persist for decades. Your DNA doesn't change, but how it's read does. And that can affect everything from your risk of depression to your susceptibility to autoimmune diseases. Diet also changes epigenetic markers. Folate, found in leafy greens, is a key donor of methyl groups. If your diet is deficient in folate, methylation patterns shift. Genes that should be silenced might become active. Genes that should be active might get silenced. This is why maternal nutrition during pregnancy is so critical. The fetus's developing cells are laying down epigenetic patterns that will last a lifetime. What the mother eats, how much stress she experiences, even air pollution exposure, all of it influences the child's epigenome. One of the most famous studies on epigenetics involves the Dutch hunger winter of 1944 to 1945. Near the end of World War II, the Nazis blocked food supplies to the Western Netherlands. Thousands of people starved. Pregnant women gave birth to underweight babies, which wasn't surprising. But decades later, researchers noticed something strange. The children born during the famine had higher rates of obesity, diabetes, and cardiovascular disease. And so did their children. The grandchildren of famine victims, who were never themselves starved, carried metabolic changes linked to their grandparents' trauma. The mechanism seems to be epigenetic. During fetal development, if the mother is starving, the fetus's body makes a prediction. Resources are scarce. Better to develop a metabolism that hoards every calorie. Epigenetic markers shift to prioritize fat storage and slow metabolism. This is adaptive if you're born into a world of famine. But if you're born into a world of plenty, those same adaptations become risk factors for metabolic disease. And those markers can persist across generations, even if the children and grandchildren never experience hunger themselves. Holocaust survivors show similar patterns. Studies have found altered methylation in genes related to stress response in survivors and their children. The trauma didn't just affect those who lived through it. It left a biological signature that was passed down. This isn't genetic inheritance in the traditional sense. The DNA sequence didn't change, but the epigenetic instructions sitting on top of that DNA did. And those instructions, in some cases, were transmitted to the next generation. How this happens is still being researched. Epigenetic markers are supposed to be erased and reset between generations. During the formation of eggs and sperm, most methylation patterns are wiped clean so the next generation starts fresh. But not all markers are erased. Some slip through. And certain experiences, severe stress, trauma, malnutrition, seem to leave marks resilient enough to survive the reprogramming process. It's as if evolution has created a way for organisms to pass on information about environmental conditions without changing the genetic code itself. This has profound implications. It means your lifestyle choices don't just affect you, they might affect your children and grandchildren. If you smoke, if you're chronically stressed, if you eat poorly, you're potentially altering epigenetic patterns in your germ cells. And those altered patterns could influence the health of descendants you'll never meet. You're not just living your own life, you're shaping the biology of future generations. There's also evidence that positive experiences can change epigenetics. Meditation, exercise, strong social connections, All of these have been associated with beneficial epigenetic changes. Reduced inflammation markers, improved stress response, even changes in telomerase activity, the enzyme that rebuilds telomeres. You can, to some extent, rewrite your epigenetic story. The marks left by trauma or poor environment aren't necessarily permanent, but reversing them takes effort, time, and often significant lifestyle changes. Epigenetics also plays a role in aging. As you get older, your methylation patterns drift. Some genes that should be on get turned off, others that should be off get turned on. This epigenetic drift is so predictable that scientists can estimate your biological age just by looking at methylation patterns in your DNA. It's called the epigenetic clock. And interestingly, some people's epigenetic age is younger than their chronological age. Others are older. Lifestyle factors like diet, exercise, and stress management seem to influence how fast your epigenetic clock ticks. Cancer is also deeply tied to epigenetics. Many cancers involve not just genetic mutations, but also abnormal methylation patterns. Tumor suppressor genes get hypermethylated and silenced. Oncogenes, genes that promote cell division, get hypermethylated and activated. The cancer cell's epigenome becomes chaotic. And because epigenetic changes are potentially reversible, unlike genetic mutations, there's hope that drugs targeting epigenetic machinery could treat cancer. Some are already in use. Azacitidine and decitabine are drugs that inhibit DNA methylation and are used to treat certain blood cancers. Identical twins offer another window into epigenetics. Twins share the same DNA sequence, but as they age, their epigenomes diverge. One twin might develop an autoimmune disease while the other doesn't. One might be more prone to anxiety. The differences aren't genetic. They're epigenetic, shaped by different experiences, environments, and random epigenetic drift over time. This explains why identical twins, despite having identical genomes, are not identical people. What's philosophically disturbing about epigenetics is the erosion of the boundary between nature and nurture. Your genes aren't fixed destiny. Your environment isn't just external influence. The two are in constant conversation, and that conversation is written in chemical marks on your DNA. You inherit not just genes, but also the epigenetic consequences of your ancestors' lives. You're carrying their struggles, their adaptations, and their survival strategies in the margins of your genome. And this raises uncomfortable questions. If trauma can be inherited epigenetically, what does that mean for justice, for healing, for understanding inequality? Communities that have experienced generations of oppression, poverty, or violence might be carrying biological scars that make recovery harder. It's not just social or psychological, it's molecular. And it doesn't excuse the systems that cause that trauma, but it does mean the damage runs deeper and lasts longer than we thought. So, the next time someone says you are what your genes made you, remember that's not the whole story. You're also what your environment made your genes express. You're the sum of your ancestors' experiences written in methyl groups and histone modification. You're carrying forward not just DNA, but annotations, instructions, and warnings from people who came before you. Your genome is a text, but your epigenome is the commentary, and both are shaping who you are right now. Evolution is supposed to be slow, gradual changes accumulating over generations through mutation and natural selection. A tree branching upward through time, each species inheriting traits only from its ancestors. That's the story you learned in school. But it's not the whole story, because in the microbial world, Evolution doesn't wait for reproduction. Bacteria don't just inherit genes from their parents. They trade them, they steal them. They pick them up from the environment like finding cash on the sidewalk. And this process, called horizontal gene transfer, has rewritten the tree of life into something more like a tangled web. And it's happening inside you right now. Horizontal gene transfer is exactly what it sounds like. Genes moving sideways between organisms, not just down through generations. In bacteria, this is routine. A bacterium can acquire a new gene from a completely unrelated species and start using it immediately. No need to wait for offspring, no need for sexual reproduction, just direct genetic exchange. And the implications are staggering, especially when it comes to antibiotic resistance. Let's say you take an antibiotic to treat a bacterial infection. Most of the bacteria die, but a few survive because they happen to carry a gene that makes them resistant. Maybe that gene codes for an enzyme that breaks down the antibiotic, or a pump that ejects it from the cell, or a mutation in the antibiotic's target that prevents it from binding. That resistant bacterium survives and multiplies. So far, this is just natural selection, but here's where it gets worse. That resistant bacterium doesn't keep the resistance gene to itself. It can transfer it to other bacteria through several mechanisms. The most common is a plasmid, a small circular piece of DNA separate from the main chromosome. Plasmids carry extra genes, often including antibiotic resistance. Bacteria can copy plasmids and pass them to neighboring bacteria through a process called conjugation. One bacterium extends a tube-like structure called a pilus, connects to another bacterium, and physically transfers the plasmid. It's like handing someone a flash drive full of genetic instructions. This means antibiotic resistance can spread through a bacterial population in hours, not generations. A resistant strain of E. coli in your gut can transfer its resistance genes to Salmonella, Shigella, or other species you might encounter. The resistance isn't confined to one lineage. It jumps across species boundaries as if those boundaries don't exist. And they don't, not for bacteria. Species is almost a meaningless concept in the microbial world because genes flow so freely between them. There's also transformation, where bacteria pick up DNA directly from their environment. When a bacterium dies, its DNA spills out into the surroundings. Other bacteria can absorb those naked DNA fragments and incorporate them into their own genomes. If that DNA contains a useful gene, like antibiotic resistance, the bacterium now has it. This is how researchers first discovered DNA was the genetic material. They watched bacteria transform by absorbing DNA from dead cells. Then there's transduction, where viruses act as genetic couriers. Bacteriophages, viruses that infect bacteria, sometimes accidentally package bits of the host bacterium's DNA when they replicate. When the virus infects a new bacterium, it injects that stolen DNA along with its own. The new host now has genes from a completely different bacterium. Viruses are inadvertently shuffling genes between bacterial species like a microscopic genetic postal service. The speed of horizontal gene transfer is terrifying when you consider antibiotic resistance. In 1928, penicillin was discovered, and it was a miracle. Bacterial infections that had been death sentences became treatable. But by the 1940s, penicillin-resistant bacteria were already appearing. Today, we have bacteria resistant to nearly every antibiotic we've developed. Methicillin-resistant Staphylococcus aureus, or MRSA. Carbapenem-resistant Enterobacteriaceae, or CRE. Extensively drug-resistant tuberculosis. These aren't mutants that evolved resistance slowly over centuries. They're bacteria that acquired resistance genes from other bacteria, sometimes multiple resistance genes at once, through horizontal gene transfer. And it's not just happening in hospitals. Antibiotic resistance genes are spreading in the environment. Farms use antibiotics in livestock to promote growth and prevent disease. Those antibiotics create selective pressure. Resistant bacteria thrive in the guts of animals, in manure, in soil, in water runoff. They mingle with bacteria from wild animals, from humans, from other farms. Resistance genes spread through this vast microbial network. A gene that originated in a pig farm in China can end up in a human pathogen in Europe within years. Even more unsettling, some resistance genes are ancient. They predate the invention of antibiotics. Scientists have found antibiotic resistance genes in bacteria from permafrost samples 30,000 years old. These genes evolved naturally, probably as defense mechanisms against antibiotic compounds produced by soil, fungi, and bacteria. But when humans started mass-producing antibiotics, we created enormous selective pressure. Those ancient genes, buried in rare bacteria, suddenly became valuable. Horizontal gene transfer mobilized them, spreading them rapidly through bacterial populations worldwide. Now here's the really disturbing part. You carry trillions of bacteria in your gut, on your skin, in your mouth. Many of them harbor resistance genes. Even if you've never taken antibiotics yourself, you're probably carrying resistant bacteria just from environmental exposure. And those bacteria are constantly swapping genes. Your gut microbiome is a genetic marketplace where bacteria trade DNA like currency. If a pathogenic bacterium enters that ecosystem, it can potentially pick up resistance genes from your own commensal bacteria. Your microbiome becomes an incubator for superbugs. But horizontal gene transfer isn't limited to bacteria. It's also played a major role in the evolution of complex organisms, including humans. Your genome contains sequences from viruses. Roughly 8% of human DNA is derived from ancient retroviruses that infected our ancestors millions of years ago. These viruses inserted their genetic material into germ cells, eggs, or sperm, and those sequences were passed down through generations. Most are now inactive genetic fossils, but some have been co-opted for useful functions. One viral gene is essential for placenta development. Without it, mammalian pregnancy as we know it wouldn't be possible. There's also evidence that eukaryotic organisms, plants, fungi, animals, have occasionally acquired genes from bacteria through horizontal gene transfer. It's rare compared to what happens in bacteria, but it happens. Some plant genes involved in disease resistance appear to have bacterial origins. Bdelloid rotifers, tiny aquatic animals, have incorporated genes from bacteria, fungi, and plants. They're genetic mosaics, cobbled together from multiple kingdoms of life. This fundamentally changes how we understand evolution. The tree of life, with its neat branches diverging from common ancestors, is an oversimplification. At the base, where bacteria and archaea dominate, it's more like a web or a braided river. Genes flow not just vertically from parent to offspring, but horizontally between distant branches. The history of life isn't just descent with modification. It's also lateral acquisition, theft, and collaboration. And this has practical consequences. It means antibiotic resistance is not a problem we can solve by simply developing new drugs. As long as bacteria can share resistance genes, any new antibiotic will eventually face resistance. We're in an evolutionary arms race, and the enemy can share weapons faster than we can invent new ones. Some scientists are exploring alternatives. Bacteriophage therapy, using viruses to kill bacteria, CRISPR-based approaches to cut resistance genes out of bacterial genomes, drugs that block horizontal gene transfer itself, but these are still experimental. What horizontal gene transfer teaches us is that the boundaries we draw around species, around individuals, around genomes, are more porous than we'd like to believe. Genes don't respect those boundaries. They flow where selective pressure pushes them. And in a world saturated with antibiotics, selective pressure is pushing resistance genes everywhere. Into hospitals, farms, sewage systems, rivers, oceans, into the bacteria living inside you right now. So, when you take an antibiotic, you're not just treating an infection. You're applying selective pressure to trillions of bacteria in and around you. You're participating in a global evolutionary experiment where resistance genes are the currency and horizontal gene transfer is the mechanism. And the outcome of that experiment is uncertain. But one thing is clear: evolution isn't just slow and gradual. Sometimes it's fast, lateral, and utterly indifferent to the species barriers we thought were fixed. Life finds a way. And sometimes that way is sideways. Inside your body right now, there are cells that have stopped dividing but refuse to die. They're stuck in a kind of biological limbo, neither fully alive nor properly dead. They can't reproduce. They can't contribute to tissue repair, but they remain metabolically active, stubbornly clinging to existence while secreting a toxic cocktail of inflammatory chemicals that damage everything around them. They're called senescent cells, and they're one of the primary drivers of aging. You're accumulating them right now, slowly, invisibly, and they're poisoning you from within. Senescent cells are what happens when cells hit a crisis they can't resolve. Maybe their telomeres have become critically short. Maybe their DNA is too damaged to repair. Maybe they've been exposed to stress, radiation, or toxins that push them past the breaking point. Normally, cells in this situation have two options: repair the damage and keep going, or activate apoptosis and die cleanly. But senescent cells choose a third option. They stop dividing, but they don't die. They just sit there, occupying space, consuming resources, and causing trouble. The decision to become senescent is actually a protective mechanism, at least initially. If a cell has DNA damage but can still function, allowing it to divide could lead to cancer. So the cell slams on the brakes. It enters a permanent state of growth arrest called senescence. This prevents the damaged cell from becoming a tumor. In that sense, senescence is your body's way of prioritizing cancer prevention over tissue health. Better to have a few dysfunctional cells than a malignant tumor. And when you're young, this system works. Senescent cells appear occasionally, but they're quickly cleared away by your immune system. Macrophages and natural killer cells recognize senescent cells and destroy them. The turnover is efficient. Damaged cells are removed, healthy cells take their place, and everything stays balanced. But as you age, two things go wrong. First, more cells become senescent because they've accumulated more damage over time. Second, your immune system gets worse at clearing them. The result? Senescent cells start to accumulate. By the time you reach old age, a small but significant percentage of your cells are senescent. They cluster in certain tissues, particularly in the skin, liver, fat tissue, and blood vessels. And here's where the real problem starts. Senescent cells don't just sit quietly. They secrete dozens of inflammatory molecules, growth factors, and enzymes that break down the extracellular matrix. This toxic mixture is called the senescence-associated secretory phenotype, or SASP, and it's devastating. The SASP creates chronic, low-level inflammation throughout your body. This inflammation damages nearby healthy cells, disrupts tissue architecture, and accelerates aging. It's like having a small fire burning continuously in your tissue. Not intense enough to notice acutely, but persistent enough to cause long-term harm. This chronic inflammation is linked to nearly every age-related disease: cardiovascular disease, diabetes, arthritis, neurodegenerative diseases, cancer. Senescent cells are one of the common threads tying all of these conditions together. In your skin, senescent cells contribute to wrinkles, sagging, and loss of elasticity. They break down collagen and prevent new collagen from forming. Your skin becomes thinner, more fragile, less able to heal from injury. In your blood vessels, senescent cells make arteries stiffer and promote plaque formation. This increases your risk of heart attack and stroke. In your joints, they drive cartilage degradation and chronic inflammation, leading to osteoarthritis. Even more disturbing, senescence can spread. The inflammatory signals from senescent cells can push neighboring healthy cells into senescence. It's contagious, not through infection, but through chemical signaling. One senescent cell can create more senescent cells around it. This creates expanding zones of dysfunction in your tissues. The damage compounds over time, accelerating the aging process. Scientists have tested this in mice. When researchers genetically engineered mice to allow selective elimination of senescent cells, the results were stunning. The mice lived longer, healthier lives. They had less age-related tissue dysfunction. Their organs stayed healthier. Their fur remained thicker. They were more active. Simply removing senescent cells was enough to delay many aspects of aging. It didn't make the mice immortal, but it extended their health span, the period of life spent in good health. This discovery has sparked enormous interest in senolytic drugs, compounds that selectively kill senescent cells. Several are in development or early-stage human trials. Dasatinib and quercetin, a combination of a cancer drug and a plant compound, have shown promise in clearing senescent cells in mice and in small human studies. Fisetin, another plant-derived compound, has similar effects. These drugs don't prevent cells from becoming senescent. They just help the body clear the ones that already exist. But there are risks. Senescent cells aren't all bad. In wound healing, senescent cells help coordinate tissue repair by secreting growth factors and signaling molecules. They're temporary, normally cleared once the wound heals, but they play an important role. During pregnancy, senescent cells appear in the uterus and contribute to remodeling tissue for childbirth. Eliminating all senescent cells indiscriminately could have unintended consequences. The goal isn't to remove them entirely, but to clear the chronic, accumulated ones driving age-related disease. There's also the question of what happens to the space left behind when senescent cells are removed. Do healthy cells move in and regenerate the tissue? Or does the tissue simply collapse, creating structural problems? Early research suggests that in many tissues, clearing senescent cells allows healthy cells to proliferate and restore function. But in others, particularly in the brain, it's less clear. Neurons don't regenerate easily. Removing senescent support cells without replacing them could cause more harm than good. Senescent cells also accumulate in fat tissue, and this has metabolic consequences. Senescent fat cells, or adipocytes, secrete inflammatory molecules that interfere with insulin signaling. This contributes to insulin resistance, weight gain, and type 2 diabetes. In obese individuals, the number of senescent cells in fat tissue is significantly higher than in lean individuals. It's a vicious cycle. Obesity promotes cellular senescence, which promotes inflammation, which worsens metabolic dysfunction, which leads to more obesity. Interestingly, caloric restriction, eating significantly fewer calories while maintaining nutrition, reduces the accumulation of senescent cells. It also activates autophagy, which we'll discuss more in the next chapter, a process where cells break down and recycle damaged components. Exercise has similar effects. Physical activity improves immune function, helping the body clear senescent cells more efficiently. These interventions don't eliminate senescence, but they slow its accumulation. There's also research into preventing cells from becoming senescent in the first place. If you can reduce DNA damage, oxidative stress, and telomere shortening, you reduce the triggers that push cells into senescence. Antioxidants, at least in theory, should help. But clinical trials with antioxidant supplements have been disappointing. The body's own antioxidant systems are far more effective than anything you can swallow in pill form. And excessive antioxidant supplementation can actually interfere with beneficial oxidative signaling. What's clear is that senescent cells represent a fundamental trade-off in biology. In the short term, senescence prevents cancer. It's a safety valve that stops damaged cells from dividing out of control. But in the long term, senescent cells accumulate and drive aging. Evolution favored a strategy that protects you during your reproductive years, even if it means you'll suffer later. Natural selection doesn't care about your health at 70. It cares about your ability to reproduce at 25. Senescence is adaptive when you're young and maladaptive when you're old. So inside your tissues, right now, there are zombie cells. Not dead, not alive, just existing in a toxic stasis. They're secreting chemicals that age you faster. They're recruiting neighboring cells into the same dysfunction. They're stealing resources and giving nothing back. Your immune system is trying to clear them, but it's fighting a losing battle. Every year, more senescent cells accumulate. Every year, the inflammation gets a little worse. Every year, your tissues function a little less efficiently. And the cruelest part? You can't feel it happening. Senescence is silent. You don't wake up one day and think, yes, more of my cells have become senescent. You just notice, gradually, that you're slower, stiffer, more tired, that wounds take longer to heal, that your skin looks older, that your joints ache. All of it, driven in part by cells that should have died years ago but didn't. Cells that are clinging to existence while dragging you down with them. Senescent cells are neither alive nor dead, but somewhere in between, a biological purgatory where they wait, accumulate, and slowly poison the body that once nurtured them. And every day you age, there are more of them. Your cells have a memory. Not the kind of memory stored in your brain, but a cellular memory embedded in their biology. They remember how many times they've divided. They keep count, silently, invisibly, with no conscious awareness, but with perfect accuracy. And when they reach a certain number, somewhere between 50 and 70 divisions, depending on the cell type, they stop permanently, no matter how healthy they are. No matter how perfect the conditions, they've hit their limit, and that limit is hardwired into their structure. It's called the Hayflick limit, and it's one of the most fundamental constraints on life itself. The discovery came from Leonard Hayflick in 1961. Before his work, scientists believed that cells could divide indefinitely given the right nutrients and environment. The prevailing theory was that aging happened to the organism but not to individual cells. Cells, in isolation, were thought to be potentially immortal. Hayflick proved them wrong. He cultured human fibroblasts, cells from connective tissue, and watched them divide. They grew enthusiastically at first, doubling regularly, but around 50 divisions they slowed down, and then they stopped. They didn't die. They just entered a state of permanent growth arrest, senescence. Hayflick tried everything to restart them. He gave them fresh nutrients. He changed the culture medium. He adjusted temperature, oxygen, and pH. Nothing worked. The cells had reached their limit and would not divide again. Even more fascinating, he froze some cells midway through their divisions, stored them for years, then thawed them and allowed them to grow. The cells didn't restart from zero. They resumed counting from where they'd left off. Somehow, they remembered. A cell that had divided 30 times before freezing would divide only 20 more times after thawing. The count was preserved. This cellular memory lives in the telomeres, those repetitive DNA sequences at the ends of chromosomes we discussed earlier. Each time a cell divides, its telomeres shorten, and the cell tracks this shortening. When telomeres reach a critically short length, they trigger a DNA damage response. The cell interprets short telomeres as broken chromosomes, a catastrophic problem, and the cell's response is to stop dividing. It enters senescence to avoid the risk of passing on damaged DNA. The Hayflick limit is really a telomere limit. The countdown is molecular, but the memory is structural. But here's the disturbing part. This limit applies to almost all of your cells. Skin cells, immune cells, liver cells, lung cells. They all have a division quota, and once they've used it up, they can't regenerate. Your tissues lose their ability to repair themselves. Wounds heal slower because there aren't enough functional cells left to close the gap. Your immune system weakens because it can't produce enough new white blood cells. Your organs gradually decline because the cells maintaining them are hitting their limit. The Hayflick limit is why aging accelerates with time. When you're young, most of your cells are nowhere near their limit. They have plenty of divisions left. Tissue damage is easily repaired, but as you age, more and more cells approach or reach their limit. The reserve capacity shrinks. Eventually, you're running a body on cells that are maxed out, exhausted, barely functional. It's like running a factory where the machines are all decades old and can't be replaced. Production slows. Errors accumulate. Breakdowns become frequent, and eventually, the whole system fails. Different cell types have different Hayflick limits. Fibroblasts typically divide 50 to 70 times. Epithelial cells, which line your gut and skin, might have a slightly different range. But all normal human cells, with a few exceptions, have a finite division potential. This is why injuries in elderly people heal so slowly. The cells needed to close wounds and regenerate tissue have hit their limit. They're senescent, stuck, unable to contribute. The body tries to compensate, but it's fighting biology itself. The exceptions are important. Germ cells, the cells that become sperm and eggs, don't have a Hayflick limit. They express telomerase, the enzyme that rebuilds telomeres. This makes sense evolutionarily. If germ cells had shortened telomeres, each generation would start with shorter telomeres than the last. Within a few generations, Telomeres would be so short that embryos couldn't develop, so germ cells reset the clock. They maintain their telomeres, ensuring that each new generation starts fresh. Stem cells also have higher telomerase activity than most cells. Stem cells need to divide many times throughout your life to replace damaged tissues. If they hit the Hayflick limit too early, you'd run out of regenerative capacity. So, stem cells have a higher division quota, but even stem cells aren't immortal. Their telomerase activity slows with age. Their telomeres still shorten, just more slowly. And eventually, even stem cells senesce. That's why older people have fewer functional stem cells and less regenerative capacity. And then there are cancer cells. Most cancer cells reactivate telomerase or use an alternative mechanism to maintain telomeres. They bypass the Hayflick limit entirely. This is one of the hallmarks of cancer. Normal cells are constrained by their division quota. Cancer cells are not. They divide indefinitely, which is why tumors grow uncontrollably. The Hayflick limit is one of your body's primary defenses against cancer. It ensures that even if a cell starts dividing inappropriately, it will eventually run out of steam and stop. But cancer cells have evolved ways around this defense. The healer cells we discussed in Chapter 1 are a perfect example. Henrietta Lacks' cancer cells activated telomerase, allowing them to divide indefinitely. Decades after her death, those cells are still growing in labs worldwide. They've divided far more than 50 or 70 times. They've divided thousands, maybe tens of thousands of times. They're biologically immortal because they've escaped the Hayflick limit. But that immortality came at the cost of the organism. The cells that can live forever killed the body they came from. Researchers have tried to extend the Hayflick limit experimentally by artificially expressing telomerase in normal human cells. Scientists can make those cells divide far beyond their natural limit. Some have divided hundreds of times without entering senescence. But there's a catch. Cells with artificially extended lifespans are more prone to accumulating mutations. The longer a cell lives, the more chances it has to develop errors. Over enough divisions, even with telomerase, cells start to malfunction. The Hayflick limit isn't just arbitrary. It's a balance between regeneration and cancer risk. There's also evidence that the Hayflick limit is influenced by external factors. Cells exposed to chronic stress, oxidative damage, or inflammation hit their limit faster. Their telomeres shorten more quickly. Conversely, cells in a healthy, low-stress environment may divide more times before senescence. This is one reason why lifestyle factors like diet, exercise, and stress management affect aging. They don't change the fundamental mechanism, but they influence how quickly you reach the limit. In some ways, the Hayflick limit is a timer on your life. Every cell division burns through a finite resource. And once that resource is depleted, the cell stops contributing. You are, at the cellular level, running on borrowed time. Your tissues are maintained by cells that are slowly counting down to zero. And when enough of them hit zero, your body can no longer sustain itself. Organs fail, systems break down, and eventually you die. Not because something went wrong, but because everything went exactly as biology designed it. What's particularly sobering is that the Hayflick limit applies even in ideal conditions. Even if you never get sick, never have an injury, never experience stress, your cells will still hit their division limit. Aging isn't just accumulated damage. It's a built-in feature. Your cells are programmed to stop, and no amount of healthy living can override that programming. You can slow it down, optimize it, delay it, but you can't escape it. The Hayflick limit also explains why cloning is so difficult. When you clone an animal, you're starting with a cell from an adult organism. That cell has already used up some of its divisions. Its telomeres are already shortened, so the cloned animal starts life with older cells. Dolly the sheep, the first cloned mammal, developed arthritis and other age-related conditions earlier than normal sheep. She was born old, at least at the cellular level, because her cells inherited the division history of the adult sheep she was cloned from. So your cells are keeping score. They're counting every division, tracking every shortening of the telomeres, and when the count reaches the limit, they stop. It's precise. It's inexorable. It's a biological countdown that started the moment you were conceived, and there's no pause button, no reset. Just a slow, inevitable march toward the moment when too many of your cells have hit their limit and your body can no longer function. That's the Hayflick limit. Not a disease, not a failure, just the price of being mortal. When resources run low, when food becomes scarce, when survival is uncertain, your body doesn't just shut down and wait for better times. It does something far more primal. It starts eating itself, not in a destructive, desperate way, but methodically, strategically, breaking down its own components to stay alive. This process is called autophagy, literally self-eating, and it's happening inside your cells right now. Your body is constantly deciding which parts of itself to consume, which proteins to dismantle, which organelles to sacrifice. It's cellular cannibalism in the name of survival, and it's absolutely essential. Autophagy is one of the most fundamental recycling systems in biology. Your cells are packed with proteins, organelles, and molecular machinery. Over time, these components wear out, proteins misfold, mitochondria become damaged, cellular debris accumulates. If left unchecked, this junk would clog the cell, interfere with normal function, and eventually kill it. Autophagy is how cells clean house. They wrap damaged components in a membrane, forming a structure called an autophagosome. This autophagosome then fuses with a lysosome, an organelle filled with digestive enzymes. The contents are broken down into basic building blocks, amino acids, lipids, sugars, and those building blocks are recycled to make new proteins and structures. It's elegant, it's efficient, and it's happening constantly at a low baseline level in almost every cell in your body. But autophagy ramps up dramatically under stress. When you're fasting, when nutrients are scarce, your cells need energy and raw materials. They can't get them from food, so they turn inward. Autophagy increases. The cell starts breaking down non-essential proteins, damaged mitochondria, and other components it can afford to lose. It's a survival mechanism, allowing the cell to persist through lean times by consuming itself. This isn't as grim as it sounds. Autophagy is selective. Cells don't randomly cannibalize themselves. They target damaged, dysfunctional, or unnecessary components. A mitochondrion that's leaking reactive oxygen species and poisoning the cell. Autophagy wraps it up and breaks it down. A protein aggregate that's starting to clog cellular machinery. Autophagy clears it. In this way, autophagy is quality control. It removes the broken parts so the cell can keep functioning with what's left. Autophagy is also critical for longevity. Studies in yeast, worms, flies, and mice have consistently shown that increased autophagy extends lifespan. When researchers genetically enhance autophagy, organisms live longer and stay healthier. When they block autophagy, organisms age faster and die younger. The connection is clear. Efficient cellular recycling and cleaning prolongs life. Autophagy keeps cells young by preventing the accumulation of damaged components that drive aging. In humans, autophagy declines with age. Your cells become less efficient at clearing out junk. Damaged proteins accumulate. Dysfunctional mitochondria linger. This contributes to age-related diseases. In neurodegenerative conditions like Alzheimer's, Parkinson's, and Huntington's disease, autophagy is impaired. Misfolded proteins build up in neurons, forming toxic aggregates. The cell's recycling system can't keep up. The aggregates grow, interfere with neuronal function, and eventually kill the cell. Enhancing autophagy, at least in animal models, can reduce these aggregates and slow disease progression. Fasting is one of the most powerful triggers of autophagy. When you stop eating for an extended period, insulin levels drop and glucagon rises. This hormonal shift signals to your cells that nutrients are scarce. In response, autophagy is activated. Cells start breaking down stored components to generate energy and building blocks. This is why intermittent fasting and extended fasting have gained attention for their potential health benefits. They're not just reducing calorie intake, they're activating a deeply conserved cellular cleaning process. Exercise also stimulates autophagy, particularly in muscle cells. When you exercise, your muscles experience stress, energy demands increase, cellular components become damaged, autophagy ramps up to clear the damage and recycle materials. This is part of why exercise is so beneficial. It's not just about burning calories or building strength. It's about triggering cellular maintenance programs that keep your tissues healthy and functional. Sleep is another time when autophagy increases, especially in the brain. During sleep, your brain clears out metabolic waste and damaged proteins that accumulated during waking hours. The glymphatic system, a waste clearance pathway in the brain, is most active during sleep, and autophagy plays a supporting role. Neurons ramp up their recycling processes, breaking down damaged components while the brain is relatively quiet. This is one reason why chronic sleep deprivation is so harmful. You're not just tired, you're preventing your brain from cleaning itself. Waste products accumulate, increasing the risk of neurodegenerative disease over time. But autophagy is a double-edged sword. Too much can be harmful. If autophagy is overactive, cells can break down essential components, weakening themselves to the point of death. This happens in certain muscle-wasting diseases where excessive autophagy contributes to muscle loss. Cancer cells also exploit autophagy. When tumors are deprived of nutrients or treated with chemotherapy, they activate autophagy to survive. They cannibalize themselves just enough to stay alive until conditions improve. This makes some cancers harder to treat. Blocking autophagy in cancer cells can make them more vulnerable to therapy, but it also risks harming normal cells that rely on autophagy for maintenance. There's also a specific form of autophagy called mitophagy, where cells selectively remove damaged mitochondria. Mitochondria are the power plants of the cell, but when they malfunction, they leak reactive oxygen species that damage DNA and proteins. Mitophagy identifies these rogue mitochondria, tags them for destruction, and recycles their components. This quality control is essential for preventing oxidative stress and maintaining cellular energy production. When mitophagy fails, damaged mitochondria accumulate, contributing to aging, neurodegenerative diseases, and metabolic disorders. Researchers are exploring drugs that activate autophagy as potential anti-aging therapies. Rapamycin, An immunosuppressant drug inhibits a protein called mTOR, which normally suppresses autophagy. By blocking mTOR, rapamycin activates autophagy. In animal studies, rapamycin extends lifespan and delays age-related diseases. It's one of the most promising longevity interventions discovered so far. But it has side effects, including immune suppression, which limits its use in healthy people. Scientists are working on developing safer compounds that can activate autophagy without the downsides. Spermidine, a compound found in foods like wheat germ, soybeans, and aged cheese, also promotes autophagy. Studies show that dietary spermidine can extend lifespan in yeast, flies, worms, and mice. Human trials are ongoing, but early evidence suggests it might improve cardiovascular health and cognitive function. The mechanism isn't fully understood, but spermidine seems to enhance cellular recycling and reduce age-related damage. What's philosophically striking about autophagy is that it reveals a fundamental truth about survival. Life requires sacrifice. To keep going, your cells must consume parts of themselves. They must choose what to preserve and what to dismantle. It's a constant negotiation between survival and self-destruction. Too little autophagy, and you drown in your own waste. Too much, and you consume yourself into oblivion. The balance is everything. Autophagy also challenges the idea of a stable, unchanging self. At the cellular level, you're not a fixed entity. You're a dynamic process. Your proteins are constantly being broken down and rebuilt. Your organelles are constantly being recycled and replaced. The molecules that made up your body a year ago are mostly gone, digested by autophagy and replaced with new ones. You're not the same physical structure you were yesterday. You're a pattern maintained by continuous destruction and renewal. And this process is ancient. Autophagy exists in yeast, in plants, in bacteria, in every domain of life. It evolved billions of years ago as a way for cells to survive starvation, but it's been co-opted for so many other functions: quality control, immunity, development, aging. It's one of the most versatile and essential processes in biology. Without it, multicellular life as we know it wouldn't exist. So when you fast, when you exercise, when you sleep, you're not just resting or abstaining from food. You're activating an ancient program that tells your cells to clean house, to break down the damaged parts, to recycle what can be saved. You're triggering self-consumption in the service of survival. Your body is eating itself carefully, selectively, constantly, and that's not a sign of failure, it's a sign of health. Because life doesn't just build, it also dismantles. And knowing when to do which is what keeps you alive. We've spent the last few hours together exploring some of the darkest corners of biology. Immortal cells that outlived their host, bacteria outnumbering your own cells and potentially controlling your thoughts, parasites rewiring your brain, cancer as evolution turned inward, proteins that corrupt themselves without being alive, cells that refuse to die but poison everything around them, timers counting down your divisions, your body eating itself to survive. It's been unsettling, disturbing even. But now, as we near the end of this journey, it's time to ask the question that ties all of this together. Why does any of this matter? Because understanding dark biology isn't about dwelling in fear or surrendering to inevitability. It's about seeing life as it actually is, not as we wish it were. The biology we've explored tonight doesn't fit neatly into the sanitized narratives of textbooks or the optimistic promises of wellness culture. It's messy, it's contradictory, it's often unsettling, but it's real. And confronting that reality, really sitting with the discomfort of it, is the only way to understand what it means to be alive. Take the HeLa cells. Henrietta Lacks's story is tragic, exploitative, and scientifically revolutionary all at once. Her cells shouldn't still be alive, but they are. They've contributed to medical breakthroughs that have saved millions of lives. But they also represent a violation, a reminder that progress often comes at a cost that isn't evenly distributed. Her immortality wasn't chosen, it was taken. And yet, without those cells, we'd be decades behind in understanding cancer, viruses, and cellular biology. There's no clean moral here, just complexity, just the uncomfortable truth that good and harm can emerge from the same source. Or consider your microbiome. You're not an individual, you're a colony, and that's not a metaphor. The bacteria in your gut are influencing your mood, your cravings, maybe even your personality. You like to think your decisions are yours, that you're the captain of your own ship, but you're sharing that ship with trillions of passengers, and some of them are whispering suggestions to the captain. Does that diminish your autonomy? Maybe. But it also expands your sense of self. You're not a lone organism struggling in isolation. You're part of an ecosystem, interdependent, inseparable from the life around and within you. The parasites that control behavior, the cancers that evolve inside you, the prions that spread without being alive, these aren't aberrations. They're part of the same biological systems that allow you to exist. Life is competitive, it's opportunistic, it's relentless. And sometimes the strategies that work for one organism come at the expense of another. You're not exempt from this. You benefit from it when your immune system destroys a bacterium. You suffer from it when a parasite takes hold. It's not personal. It's just biology doing what biology does, optimizing for survival without regard for fairness or comfort. Aging, too, isn't a failure. It's a design choice made by evolution. Telomeres shorten. Cells senesce. The Hayflick limit kicks in. Your body is programmed to decline because that's the trade-off evolution made to prevent cancer and allow for generational turnover. You age so your children don't inherit your accumulated mutations. You decline so the species can adapt. It's brutal on the individual level, but it works on the population level. And that tension between what's good for you and what's good for the species runs through all of biology. Apoptosis teaches us that death is built into life at every level. Your cells carry instructions for their own destruction, ready to execute themselves the moment they're no longer useful or become dangerous. It's not tragic, it's elegant. Death isn't the opposite of life, it's part of the process. Every finger you have, every space between your toes, every neuron that survived infancy, exists because other cells died to make room. You are sculpted by death as much as by growth. And autophagy reveals that survival often requires sacrifice. Your cells consume themselves to keep going. They dismantle their own components, recycle their own structures, and rebuild from the wreckage. It's not defeat, it's adaptation. Life doesn't just accumulate, it also subtracts. And knowing when to let go, when to break down the old to make room for the new, is as essential as knowing when to build. Epigenetics shows us that the past isn't past. It's written into your biology. Your grandmother's trauma, your grandfather's starvation, your parents' stress, all of it might be influencing your genes right now. You're not starting from zero. You're starting from the accumulated experiences of everyone who came before you. That's heavy. But it's also a reminder that you're part of a lineage, a chain of survival stretching back thousands of generations. You exist because your ancestors endured, and what you do now might echo in your descendants' biology. Horizontal gene transfer teaches us that boundaries are porous. Genes flow between species. Bacteria trade resistance like currency. Your genome contains viral DNA from infections millions of years ago. The tree of life is more like a web, and the rigid categories we use to organize life, species, individuals, self, are conveniences, not truths. Life is collaborative and competitive, often at the same time, and you're part of that tangled web whether you realize it or not. So what does all of this dark biology tell us? It tells us that life is fragile, resilient, contradictory, and awe-inspiring all at once. It tells us that you're not a simple machine with replaceable parts. You're an impossibly complex ecosystem, navigating trade-offs that evolution made millions of years ago. You're a colony of cells, some yours, some not. You're a collection of ancient compromises between survival and self-destruction. You're mortal by design, but conscious enough to know it. And that tension between the biological reality of what you are and the subjective experience of being you is where meaning lives. Understanding dark biology doesn't make life meaningless. It makes it more meaningful. Because when you know how precarious this all is, how many things have to go right just for you to wake up in the morning, you start to see existence itself as the miracle. You're alive despite telomeres shortening, despite senescent cells accumulating, despite parasites, cancers, and prions lurking in the background. You're alive because trillions of cells are cooperating, mostly in a project called you, and that cooperation, fragile and temporary as it is extraordinary. The darkness in biology isn't nihilistic. It's clarifying. It strips away the illusions and shows you what's actually holding you together. Not divine intervention, not cosmic justice, just chemistry, evolution, and the relentless, indifferent creativity of life finding ways to persist. And somehow, against all odds, you're here, conscious, curious, capable of understanding the very mechanisms that constrain you. That's not nothing. That's everything. So as we close this 3-hour journey through the unsettling truths of biology, remember this. You're not separate from these processes. You are these processes. You're the telomeres shortening, the cells senescing, the mitochondria failing, the autophagy recycling, the microbiome influencing. You're all of it, happening simultaneously, in a body that's both more fragile and more resilient than you ever imagined. And understanding that really sitting with the discomfort and the wonder of it is the closest thing to wisdom biology can offer. The darkness isn't something to fear, it's something to know, because only by looking directly at the uncomfortable truths can you appreciate the improbable beauty of being alive at all. You're a temporary collaboration of matter and energy, shaped by billions of years of evolution, held together by mechanisms that are slowly, inevitably failing. And yet, here you are, still breathing, still thinking, still curious enough to spend 3 hours listening to the biology that's both creating and destroying you. That's not defeat. That's defiance. And it's the most human thing there is. So here we are, 3 hours later. 3 hours of dark biology, unsettling truths, and concepts that might have genuinely changed how you see yourself. We started this journey promising to explore the shadows, the parts of biology that textbooks gloss over and wellness influencers don't want to talk about. And we did. We look directly at the uncomfortable reality of what it means to be a living organism in a universe that's indifferent to your survival. We met Henrietta Lacks, a woman who died more than 70 years ago, but whose cells are more alive now than ever. We discovered that you're outnumbered in your own body by bacteria that might be influencing your thoughts and decisions. We learned about chimerism, that you might be carrying DNA from siblings you never met or children you gave birth to decades ago. We explored telomeres, those molecular timers counting down every division until your cells simply stop. We confronted parasites that hijack your brain, cancers that are you turned against yourself, and prions that corrupt without even being alive. We sat with the reality of apoptosis, that every cell in your body carries instructions for its own death and executes them when signaled. We traced your mitochondria back to an ancient bacterial infection that became the most successful partnership in evolutionary history. We examined epigenetics, the idea that your grandmother's trauma might be written into your genes right now. We watched bacteria trade resistance genes like currency through horizontal gene transfer. We met the zombie cells, senescent and toxic, accumulating in your tissues and driving aging. We acknowledged the Hayflick limit, that cellular memory that counts every division and stops when the quota runs out. And we explored autophagy, your body's practice of eating itself to survive. These aren't just abstract concepts. This is happening inside you, right now. As you listen, your telomeres are shortening. Your senescent cells are secreting inflammatory signals. Your mitochondria are producing energy while slowly damaging themselves. Your microbiome is communicating with your brain. Your cells are undergoing autophagy, breaking down damaged components and recycling them. All of it, invisible, silent, relentless. You're not a static thing, you're a process. A controlled chaos of biological mechanisms, most of which are working to keep you alive, and some of which are slowly, inevitably, pulling you toward death. And that brings us back to why you're here tonight. Why you chose to spend 3 hours listening to the dark side of biology instead of something lighter, easier, more comforting. Maybe you came here because you were curious. Maybe because you couldn't sleep and needed something to occupy your mind. Maybe because you've always suspected that the biology you learned in school was sanitized, simplified, missing the deeper truth. Whatever brought you here, you stayed. You listen. You followed us through immortal cells, brain-controlling parasites, self-destructive proteins, and the inevitability of cellular aging. And you're still here. That says something about you. It says you're not afraid of complexity. You're not looking for easy answers or comforting platitudes. You want to understand how things actually work, even when that understanding is uncomfortable, especially when it's uncomfortable, because there's a kind of peace that comes from knowing the truth, even when the truth is that you're mortal, finite, and subject to biological processes you can't control. The paradox of dark biology is that it's simultaneously humbling and empowering. Humbling because it reminds you that you're not the autonomous, independent individual you think you are. You're a colony, a collaboration, a temporary arrangement of matter held together by ancient evolutionary bargains. You didn't choose to have telomeres that shorten, or mitochondria descended from bacteria, or a microbiome that influences your mood. You inherited all of it. It was decided for you by evolution, by your ancestors, by the basic constraints of biochemistry. But it's also empowering because understanding these mechanisms gives you agency within the constraints. You can't stop your telomeres from shortening, but you can slow the process through lifestyle choices. You can't eliminate senescent cells, but you can reduce their accumulation through exercise and nutrition. You can't rewrite your epigenetic inheritance, but you can influence which genes get expressed. You can't control your microbiome entirely, but you can shape it through diet and environment. Knowledge doesn't give you omnipotence, but it gives you options. And in a biological system as complex as a human body, options matter. There's also something strangely comforting about the darkness. When you know that aging is programmed, that cells are designed to fail, that death is built into the system, you stop fighting reality and start working with it. You stop blaming yourself for being tired, for healing slower, for not having the energy you had at 20. You're not failing. Your biology is doing exactly what it's supposed to do, and there's a kind of freedom in that acceptance. You're not broken. You're just mortal, and mortality isn't a flaw. It's the price of being a complex organism in a universe governed by entropy. Sleep, in this context, becomes even more important than you might have thought. Because sleep is when your brain clears metabolic waste through the glymphatic system. It's when autophagy ramps up in neurons, breaking down damaged proteins that accumulated during the day. It's when your body shifts resources toward repair instead of activity. Sleep isn't just rest, it's active maintenance. It's your cells getting a chance to clean house, recycle components, and prepare for another day of controlled chaos. When you don't sleep, you're not just tired, you're preventing your body from doing essential repair work. You're letting waste accumulate, damage persist, and systems degrade faster than they should. So as you drift off tonight, assuming you're listening to this before sleep, remember that you're not just shutting down, you're activating repair systems that have been honed over millions of years of evolution. Your brain is clearing out debris. Your cells are undergoing autophagy. Your immune system is surveying for damaged or abnormal cells and removing them. Sleep is when your body does some of its most important work, and understanding that, knowing what's happening at the cellular level, makes sleep feel less like lost time and more like essential biology. The concepts we explored tonight might seem dark, unsettling, even nihilistic at first glance, but they're not. They're realistic. And realism, when embraced fully, is liberating. You don't have to pretend aging isn't happening. You don't have to fight the inevitable with expensive supplements and wishful thinking. You can simply acknowledge that you're a biological system subject to biological constraints, and within those constraints, you can optimize. You can take care of the body you have, knowing its limitations, working with its mechanisms, and appreciating the extraordinary complexity that keeps you conscious and alive. Dark biology also reminds us that we're connected to our ancestors through epigenetic inheritance, connected to the microbial world through our microbiome, connected to evolutionary history through mitochondria and viral DNA in our genomes, connected to each other through shared vulnerabilities, shared constraints, and the shared experience of being mortal organisms aware of our own mortality. That awareness, that consciousness of the biological mechanisms underlying our existence, is what makes us human. We're the only species that can study our own telomeres, understand our own senescence, and contemplate the inevitability of our own death. That's both a burden and a gift. So, what do you do with this knowledge? You live differently, not recklessly, but intentionally. You take care of your body not because it's a perfect machine, but because it's a fragile, temporary, impossibly complex system that deserves respect. You sleep because your brain needs to clean itself. You move because your cells need autophagy. You manage stress because chronic cortisol alters your epigenome. You eat well because your microbiome influences your mind. You do these things not out of fear, but out of understanding. Not to live forever, but to live well for as long as you're here. And maybe, just maybe, you approach life with a little more humility and a lot more wonder, because you're not just a person. You're a walking ecosystem, a collaboration of trillions of cells, most of which are working to keep you alive, despite the fact that they're all slowly wearing out. You're mortal, finite, and subject to entropy. But you're also conscious, curious, and capable of understanding the very processes that limit you. And that paradox, that tension between biological constraint and conscious awareness, is where the beauty lives. Before you go, take a moment to like this video if you found it valuable, and subscribe to the channel if you want more content like this. But only if you genuinely enjoyed the journey we took tonight. I don't need empty clicks. I need curious minds who want to understand the world as it actually is, not as we wish it were. And let me know in the comments, which concept disturbed you the most? Was it the immortal healer cells? The brain-controlling parasites? The senescent zombie cells? Or maybe the realization that you're more bacteria than human? I'd love to know what resonated with you, what unsettled you, and what changed how you see yourself. As you close your eyes tonight, remember this. You're not perfect, you're not eternal, but you're here. And being here, being alive and aware in a universe that doesn't guarantee either, is extraordinary. So sleep well. Let your brain clear its waste. Let your cells undergo autophagy. Let your body do the repair work it's been doing for millions of years. And wake up tomorrow knowing a little more about the dark, beautiful, terrifying biology that makes you possible. Thank you for joining me on this journey through the shadows. Until next time, rest well, and remember, the darkness isn't something to fear. It's something to understand.